So welcome back. Letting people file in. Our first speaker will be Dimitri. He's going to be presenting on global physics and biogeochemical reanalyses. Excited to start this.
Thank you. Thank you, everyone. I want to thank the US CLIVAR committee for organizing this. This is my first meeting since Ocean Sciences 2020, and it's so nice to be able to touch people. I mean, it's so exciting. Also, the food has been wonderful. I don't know, I thank the caterers. And I want to thank the scientific committee also for inviting me. Michelle Garrick and Antoinette, I really appreciate it, because this is not my expertise. I was kind of surprised to be invited here. So I was asked to talk about ocean physics and biogeochemical reanalysis. Unfortunately, I can't do that. It's too much of a topic, and I'll use the excuse that I prepared my slides at the last minute. So I'm going to focus on things that I know a little bit better, work that I've been involved with. Oops, will it work? Let's see. Maybe I'll click on this one.
Yeah, try clicking it.
Yep, perfect. So obviously, this is work done with a very large number of people. There's no way that I could do that by myself, and consequently, I only know like a tiny bit of everything that that's involved in this. In terms of the contributors, I put a list there. I probably forgot a few people, but let me mention a few. Dustin Carroll and Holger Briggs are my former postdocs, and they've done a lot of the work that I will discuss today. Chris Hill, Stephanie Ditkovich, they were my office mates at MIT, so they've been incredibly helpful and involved in this work. Let me mention Michelle Garrick. She was part of this. She's part of the organizing committee. And in terms of my Blair Witch project, I put Raphael in there because I expect him to contribute very soon. So everything that I'm going to talk about is mostly from these three papers, summarizing papers. So there's a lot of details I won't have time to go into, but these are published, or email me or come and talk to me, and I'll be glad to answer questions that I can and tell you who can answer the questions that I can't. So, oops, let me try again. Man, this is a tough one. Okay, here's the outline. I want to give some motivation. Why do we want a data-constrained ocean biogeochemistry model as opposed to a free-running model? The second one I just added yesterday at the last minute. We had in one of the breakout sessions, someone mentioned GLORIS. And I wanted to, GLORIS is a fantastic product, but I wanted to talk 2 slides why it's different from ECHO and why ECHO is really the one that's useful for biogeochemical studies, while GLORIS, you cannot really use it for that because it doesn't conserve properties. I'm going to give you a brief history of ECHO Darwin. It's going to be a personal history, but it's kind of shows you how accidentally I became involved with this. I'll talk a little bit about the physics and biogeochemistry data assimilation that goes into this. I will talk about one application, so that kind of the third paper on the list, which is attribution of DIC variability to various physics, biology, air-CO2 flux, and some summary and future directions. So, why do we do this work? Well, as you know, the ocean is a huge reservoir of carbon. There's many versions of this figure that you probably have seen in the IPCC reports and various publications. But basically, the ocean contains 95% of the active carbon, the carbon that can interact with the atmospheric carbon and the land surface carbon during human lifetimes, if you like. There's of course a lot of carbon stored in rocks, but that's accessible on much slower timescales. So far the ocean has absorbed 40% approximately of the fossil fuel CO2 emissions since the beginning of the Industrial Revolution. However, the only reason that it's 40% as opposed to 95%, as would the stoichiometric ratio of the reservoirs would suggest, it's just that the ocean takes time to absorb things, right? So if everything is linear at equilibrium, you expect the ocean to absorb almost everything that we put in the atmosphere, but it's going to take some time, and maybe things are not linear. And that's why we really worry about the ocean, because small changes in the ocean absorption rate can have, so the time that it takes for the ocean to absorb the carbon that we put in the atmosphere, it can have very large consequences in the peak atmospheric CO2, and that's something we care about. So in order to better understand what the ocean has done and will do, we want to understand the mechanisms of air-sea exchange, transport, and storage of carbon by the ocean. So as an example, here is a figure that shows the global ocean CO2 sink from 1995 to 2018. And there's three types of estimates. The blue line is from data-based reconstructions like Landschucher and Roggenbeck that I think they were discussed earlier today. And you can see the average, the blue line, and that blue shading is kind of the standard deviation of all these estimates. The red is from forward simulations, like the ones that were talked at the beginning of the previous session. And those happen to be an average from 9 global carbon budget hindcast models. Red, again, is the average, and the thick bar is kind of the standard deviation. So what you can see is, unlike GCB models, Eco-Darwin assimilates physical and biogeochemical observation, and hence it is more consistent with database reconstructions. However, unlike database reconstructions, Eco-Darwin solutions can be used to explore the individual processes that control ocean carbon sequestration. And if you want to diagnose drivers of ocean carbon sources in sink, you need, it's a critical requirement if you want to try to understand the future trajectory of ocean carbon reservoir in a perturbed climate system. One day I will figure out how this works. Okay, so I want to say two slides on what's the difference between echo and so-called ocean reanalysis. Actually, the methodologies that we use are sufficiently different that Carl Wunsch, who's kind of the father of echo, if you want, asked us to call it a different thing. He said, don't call it ocean reanalysis because people will assume that it's like the same methodology as applied to atmospheric reanalysis or numerical weather, where you take a numerical weather prediction system and you apply it without changing the system over a long period of time. But of course, it's some sort of filter approach, Kalman filter, where you move forward, you change the state, and you don't really have an explanation how the state has changed. And that's how the glorious ocean reanalysis works, and it's a very good system if you're trying to get really close to the observations, but you don't care about the why of this is happening. And especially if you're trying to drive an ocean biogeochemistry model, you need to have conserved quantities. You can't suddenly say a bunch of heat appeared here and a bunch of fresh water appeared here. You really need to follow and know how that happens. So here is a little schematic. I mean, we show it all the time. Again, it's a schematic. I mean, the people who use Kalman filters, like the Seek filter used by Glorious, they may, I don't know exactly the details, they may try to smooth some of these discontinuities, and the discontinuities can be small at any one time. Nevertheless, they're there. So basically here, what you have in red is kind of how traditional ocean reanalysis is done. The triangles are the observations. and you can see that your model is going forward. It ends up at X, and there you make a correction using your Kalman filter or whatever method you have, and you get closer to the observations, and then you let the model evolve again. By way of contrast, what we do in ECHO, and I'll call it ocean state estimation as opposed to ocean reanalysis, although really they're solving kind of the same problem. We do not change the prognostic variables of the model. Instead, we change the boundary conditions, the surface boundary conditions, the initial conditions, and we also adjust subgrid scale parameterizations like vertical mixing and isopycnal coefficients. And therefore, we get a solution that conserves properties as it moves along. So, for example, in the solutions that we make available right now, they span 1992 to present. We're going to extend them back in time, but right now it's 92 to present. And it's one continuous solution. It would be like having a free-running model, except that you have adjusted the boundary conditions, surface initial, and mixing coefficients so that you get closer to the observations. Now, in the past, this meant that we could not get as close to the observations as a so-called traditional ocean reanalysis. We're doing better now because we are adjusting the subgrid scale parameterizations. So we get rid of a lot of these model errors that would not allow us to get as close to observations. So, a brief history of Echo Darwin from a very personal perspective. Echo started more than 20 years ago. Actually, I was involved in Echo 5 years before that as a postdoc at MIT with Carl Wunsch, or 6 years before that. But officially, Echo started as an NOPP-funded project in 1999. The PI was Detlef Stammer at Scripps. And shortly after that, Galen McKinley asked me, can I provide her with a Echo solution so that she can do some equatorial carbon and oxygen work? And I said, sure, I'll give you our best solution, which was a approximate Kalman filter solution. She took it, and after a couple of weeks, she came back. She said, this solution sucks. I get this huge outgassing here. What's going on? And we were scratching our head for a while, and eventually I said, Galen, do you want to try our smooth solution, which is a solution where you propagate the errors back in time? And she tried that, and she goes, wow, that really fits the observation so much better. So this is, we found out the importance of property conserving ocean state estimates for driving ocean biogeochemistry. I put 2005, but plus or minus 2 years. I was invited by Nikki Groover, who was at UCLA at the time. I was at JPL, to compute tracer Green's functions in a very old Echo solution, the one that was available at the time, to participate in the so-called Augmented to ocean carbon model intercomparison study. And lo and behold, to my surprise, and not only they used it to estimate the time invariant ocean uptake of carbon, storage, and transport, and both for anthropogenic and for natural carbon. And lo and behold, they had some measures of quality of these Green's functions that had to do with comparisons with CFCs and bomb carbon. And it turned out that Echo had comparable or better skill than models that were specifically tuned for ocean biogeochemistry. For example, Princeton submitted two different models to that. And I think one of them, it was pretty comparable to Echo, but the fact that we take this solution that was off the shelf and way worse than it is today, and bingo, right away we end up with tracer Green's functions that are very competitive with models that had been specifically tuned for ocean carbon cycle studies. I thought, wow, that's cool. At the same time, Mick Follow, Stephanie Ditkovich at MIT had started the so-called Darwin project. And this animation, you may have seen it at the bottom, is a really cool, very cool, movie of the Darwin ecosystem model. And I was starting with Chris Hill. We were starting this 1/6 of a degree eddying eco solution. And in 2010, we were told, well, why, you know, there was a carbon monitoring system pilot study. Why not try to put these two things together and see what, and provide an ocean carbon biogeochemistry model for CMS. 2017, Dustin Carroll joined the group. He's the type of person I had in mind. He comes from a programming background, but interested in science. And it takes really that kind of person to move this kind of technology forward. And now, more recently, Raphael joined, and he's going to do so many amazing things. Here is kind of a picture of this Eco-Darwin system. We assimilate physics. So in green are all the satellite physics that we assimilate. Blue, the in situ observations. All those are done using the adjoint method by the ECHO project. In addition to that, we added in situ biogeochemistry constraints. They're added separately right now. So the biogeochemistry assimilation is done separately from the physics, and it's done using a low-dimensional approach at the moment, the so-called Green's functions approach. Think of it as a poor man's adjoint. Let me go a bit faster. The solution is incredibly skillful, 80, 90%, because the physics are so good. So here's a comparison of Eco-Darwin with GLODAP version 2. You can see there's a bunch of superbiomes that we defined, and you can see that we really fit the observations really well. And this is true not just for carbon, it's true for oxygen, for pH. I mean, this is kind of the reanalysis, someone earlier, it exists, it's going to get better, but we do have a reanalysis for oxygen. We haven't looked at it in detail, but it's there. And it does well, not just at the surface, but at depth. This is kind of the model data mismatch as a function of depth. It's less than 4% almost everywhere. I'm going to go quick. One application example, we used this solution to look at DIC budget terms. So DIC variability can be caused by physics, biology, and RC-CO2 flux. We can disentangle all that. I don't have time to go in detail, but it's a study that's published. If anyone is interested, can go and look. Here's one figure from that study where you basically, we did it for two different depth ranges, 0 to 100 meters and full depth. And the colors show you the importance of circulation, biology, and CO2 flux for source and sink of carbon. So I'm running out of time, so I'm just going to go to the summary slide, and some future directions. So by assimilating physical and biogeochemical observations, Eco-Darwin produces RC-CO2 fluxes that exhibit broad-scale consistency with interpolation-based products. Contrary to interpolation-based products, this solution is less sensitive to sparse and uneven observational sampling, and it permits full attribution of the inferred RC-CO2 flux spatial temporal variability, as this, this paper, for example, shows. And Eco-Darwin has been used to attribute the space-time variability of global ocean DIC to physical biological RC-CO2 flux. Some things that are underway or that we would like to do. The next big paper we'd like to publish is a detailed model data comparison. So essentially the uncertainty or the confidence quantification. And then another one where we separate. Right now we haven't yet separated anthropogenic natural carbon, and we're collaborating with Gale and McKinley to get that done. So that's underway. We have ongoing projects that aim to improve the representation of land-ocean aquatic continuum. This is why this is so perfect. And Raphael he's going to be in charge of that and has a nice poster actually shown here and in the back. We're continuing to improve the solution in many ways. As the ecophysics improves, the solution improves automatically. We're working on the ecology, on the carbon chemistry, bottom sediments, ice, biology, carbon interactions. Some science studies, some people are starting to use it for science studies, for ENSO, ocean acidification, using this model for carrying out in silico experiments for ocean carbon mitigation strategies. There's one actually proposal that just got selected for biological ocean mixing. And there's some people that are interested in using this to study kelp farming and sargassum life cycle predictions. Right now, they're just proposals, but I'll stop there. The solution, of course, is fully open, fully available. The code is clean because of Dustin Carroll. Anybody who would like to use it, please do, or ask us. However, I'll stop.
We've got about 4 minutes for questions.
Antonietta.
Just a very quick question. So it's nice to see that you are thinking about expanding along the coast and also the land particles. So what about spatial resolution? Do you have
Yes, that's going to come automatically with. So right now it's a third of a degree, so it's a horrible place to be, but it was a comfortable place because it's data constrained for the long period. We actually had a higher resolution before in the previous version, but it was shorter. But the Echo project is going for a sixth of a degree. So as the Echo solutions become available and improve, we're going to be pushing to higher resolution globally. We also are doing some studies, downscaling, regional studies where we start with boundary conditions for that. So for example, we have one in the Mackenzie River Delta, trying to learn how the river runs. One in the Gulf of Mexico for Raphael's other advisor. So downscaling is also a possibility. Yeah.
Anyone else?
Thanks, that was great. I was curious if you had any plans to apply this in a predictive mode.
Not yet. Would you like to help?
One of the ensemble Kelvin filter, one of the things that he's used is sort of ensemble for initialization. Is there a way to potentially?
We haven't started. I mean, we're kind of right now. We're still focused on getting this reanalysis, if you like, period. But certainly, eventually, if you have a really good model, it would make sense to try to apply it for prediction. And that's the advantage of a Kalman filter is the estimate you get at the end is perfect for initializing your prediction. While here, because it's the whole period, you can't be at the edge, the way that GLORIS or atmospheric reanalysis are. So that's something we'll have to think about. We haven't put any thought in it yet, but I think it would be great a couple years down the road or whenever we find a person interested to help them do that. Yeah.
So you mentioned that you adjust the subgrid scale parameters. Is that at the start of the reanalysis, or do you adjust?
So, okay, very good question. This is for the physics only. And right now, we are adjusting a time invariant, three-dimensional matrix of diapycnal, a long isopycnal, and isopycnal slumping parameters. So we have three-dimensional matrices that are being adjusted. They're adjusted over the whole period, right? Because this is using the adjoint method, and therefore you adjust over the whole period of the estimation, 1992 to kind of present. But then what we have is time invariant right now. It could become, if it's needed, it could become time varying. But right now it's time invariant.
If it is time varying, does that then change that you don't conserve?
No, of course. No, you would because what you would be adjusting is not sources and sinks of properties, but it's the mixing coefficients. So everything would still be conserved even if it was time varying, because you'd be adjusting kappa, for example, in the same way that the KPP scheme adjusts kappa at every time step. based on stability and bulk and rate.
You just have to save the trajectory of the parameters themselves as a parameter, right?
Yeah.
One last quick question.
Yeah, so before the simulations, how can you calculate for just the couple model evaluation skills?
Say that again. Before
For the data simulations. Must be very important for the predictions.
Yeah, I mean, I think that the first paper has some of that in there. We can talk offline because I've run out of time and I'm about to be booted.
Thank you. Let's give Dimitri a round of applause. And we can certainly continue the discussion at the end of the session. Next, we're going to hear from Chris Edwards, talking about regional reanalyses. Excellent compliments to Dimitri's talk.
Okay, thank you very much. I'd like to echo what, pun intended, I'd like to echo what Dimitri said. It's a real pleasure to be here. I appreciate the invitation to talk about this, and it's so nice to be in person again, so thank you, everyone. I'm here to talk about regional reanalyses, and this, I have to say, it's very similar to what Dimitri was talking about, but we fall into the reanalysis camp, so it's a slightly different flavor, but it's very similar in many ways. I'm going to talk about both physical and biogeochemical reanalyses that are relevant or operating in coastal zones, and a lot of this work gets funded by various places, but a lot of the motivation is in the IU's regional associations, and there's a lot of interest in producing state estimates or reanalyses in regional environments for a lot of coastal applications. And so on the right here is a map which shows all of the different regional associations, and everyone has some sort of modeling capability. Some of them are doing data assimilation and others are not. Here are some regional reanalysis products in different locations. So on the upper right is all of these are using the regional ocean modeling system, so that's ROMS. The one on the right is the NERACOOS one run by Rutgers, John Wilkin. The one on the far left is the PAC-IUS one that's run by Brian Powell. Down in the central middle is the one that we run at UCSC, focused on the California current. And then the one on the right is this brand new one that's called WHOI, and this is very exciting. So WHOI has been developed by Alexander Karapov, who sits at NOAA now, but was, of course, at OSU. And WHOI represents NOAA's first operational coastal assimilative model. It's now operational. As of a year ago, March 2021, and so it's being produced for a very large domain at very high resolution, 4-kilometer resolution. It's being served. It's really a fantastic product. All of these are using the same tools. They're all using ROMS, 4-dimensional variational assimilation, and I guess that's all I wanted to say. This is showing physical regions only. All right, this is almost the same figure that Dimitri showed, but I'm going to show it anyway. This is what ROMS 40var does. It's the same sort of state estimation idea, is that you've got some prior estimate, we might call it a forecast. It's in green, some data comes in, shown as the red dots, and there's some discrepancy between the forecast and the red dots that came in. So after the fact, you can go back and make adjustments to your control vector, the initial conditions, say, or the forcing or boundary conditions, and hopefully your posterior analysis, which is the blue line, is closer to the data than the green line was initially. So the only real difference between what we do and what Dimitri was describing is that we do this on a several day by several day basis, and we do adjust physics and biology without explanation. So, that's how weather forecasting works as well, and we're following that model. I apologize for showing an equation at the bottom. I have 2 or 3 of these. I'm simply showing that what data assimilation does is it minimizes a cost function, which is basically the sum of 2 terms. One term is a model data difference, and then the other term is a deviation from your background estimate, meaning you think the model is not terrible at first, so you don't want to overfit and completely deviate from your background estimate, and so you balance those 2 things. And the balancing between trying to match the data and not deviating too much from your prior estimate is embodied in these error covariance matrices, which is actually a dirty little secret in data assimilation that we don't know those actually very well. But we do our best to estimate them as much as possible. Here's a little, like others, I seem to be frozen. There we go. This is just a demonstration of what happens on a daily or a couple-day basis. We have some prior estimate. This is showing SST in our domain. Satellite flies, data comes in, and you can see some discrepancies between the prior estimate and the observations. The machinery works to produce a posterior estimate that is some combination of the prior estimate and the observations. So you can see, for example, the development of an eddy in a place that didn't exist prior. Okay, so we've been doing this for a number of years. We've been doing both historical reanalyses and near real-time state estimates for about 10 years now. The data is served on the UCSC Threads data server. Some of it is physical, some of it is biogeochemical. I'm just going to highlight, we have some historical reanalyses going back to 1980, but stopping in 2010, and then we've got another one from 1999 to 2012. They use different surface forcing, and then we have a near real-time system which has been operating. And these things are evolutionary. There are problems with them. You always try to make them better. So anyway, things change along the way. We're assimilating a lot of different data. I'll talk about that later. Okay, one reason why physical data assimilation is actually very useful is that you can find statistical relationships between the physical properties and biogeochemical data sets or fisheries data sets. And this is just an example. Mike Jaycox showed this yesterday. This is an example for fisheries management. The idea of Ecocast is that you combine a large series of observations of fish presence and absence with environmental data that you have, and you produce statistical species distribution models that could be based on model output and not solely on observed properties such as satellite temperature, in order to produce dynamic maps of habitat that can be used for management. So anyway, this is the type of thing that those historical records can be used for, and not all properties that are statistically related to the species distributions are well observed. Things like mixed layer depth or eddy kinetic energy or depth of a particular density surface. So anyway, there are, even in purely physical reanalyses, there are actually a lot of useful biogeochemical applications. Okay, so what's the data that we use for physics? This is it. It's not much more. So we have satellite altimetry, which is great, satellite temperature, which is fantastic. We've got Argo, which I can't say enough good things about. We've got gliders, which are spectacular for getting subsurface information in the coastal zone. And of course, along the coast, we've also got HF radar, which we assimilate as well. So, this is kind of it. It's a lot, but we could always use more. In the historical reanalyses, what I'm showing here are the actual number of data records that are available and can be assimilated into our regional model. Early on, 1980, well, there were always some, and it's a log scale, so there's always some in situ observations. AVHR, we had a Pathfinder, temperature estimates from satellite for a long time. Aviso started flying around 1992, Topex Poseidon, and we began to get SSH. Of course, there were other temperature sensors that came in later. Anyway, when you put it in a figure like this, you realize, all right, there really is a very finite number of pieces of information that can go into this. Nonetheless, we're very grateful to all of those pieces. I mentioned how physical estimates can be used for biogeochemical predictions, and this was another effort by Mike Jaycox when he was a postdoc. Actually, I think right after he was a postdoc, he took the historical reanalysis and he tried to anticipate the chlorophyll result in the 2015-2016 El Nino. So he looked at historical lines of the depth of the 26.0 surface for different El Nino events, and then was able to anticipate that there'd be somewhat muted response to the chlorophyll in that particular case because the preconditioning of the California current by the blob was going to alter the response of the chlorophyll. So again, sometimes physical data sets or model output can be very, very useful. The impact of the assimilation. So this figure is simply showing what happens if you take a free-running model and compare it to an assimilative model. And this particular metric is for eddy kinetic energy. So shown on the right is the original, and then shown on the left is after we assimilate. For example, sea surface height has a lot of information about eddies, and so you end up raising the energetics of the California current rather substantially. I point this out simply to say that the forward model can always be improved. There are things you can do to make it better, but there are also always limitations. A model is a model, and data helps to create more consistent environments for depending on exactly what you're looking for. Okay, this is a different topic, and this is, I think, the second and only other equation in there. So one of the neat things about this method is that you can do, we talked yesterday a little bit about OSSEs, which are observing system simulation experiments where you try to look at the value of particular observations. And you can do this with the four-dimensional variational assimilation methodology that we have where, where I can't really point very easily, sorry. The analysis is equal to the prior estimate plus some term, and the term is proportional to the model data misfit. And what that allows you to do is you can actually partition the change in your forecast associated with each and every observation. So you can calculate, for a particular metric like transport, you could say, the transport changed because this particular measurement gave us that information. And here's just some quantification of that. I'm going to just focus on the middle panel, the middle slide, which is a time series showing the contributions of different observational platforms to our state estimate for a particular metric. In this case, it's transport across 37 north between the coastline and some distance offshore and down to 500 meters. So it's upper ocean transport. And what you see is that sometimes the data makes the transport larger, sometimes the data makes it smaller, but that the different platforms contribute in a quantifiable way. Now, if you choose a different metric, you get a different relationship, but you can use this method to kind of look at your observing system as a whole and argue the value of particular data points. So we find with these sorts of studies that satellite data dominates the data that constrains our model. But if you look at the impact per data point, it's the subsurface information that actually dominates. So that argues for more things like gliders, more things like Argo floats, and so on. So anyway, I wanted to show this as a kind of an alternate way of trying to assess the observing system. Okay, in the remaining half, I would like to move on to biogeochemical data assimilation. So this is very similar to the physical data assimilation, but we do it with ecosystem models, and I'm just showing here 2 ecosystem models. We use NPZD as kind of a building tool to understand this capability and to try to develop the capability, but our workhorse is NERACOOS, which is shown on the right, which is a much more complicated one, and probably much more useful for the California current system because it has some phytoplankton diversity and zooplankton diversity. These next 2 slides are not actually about assimilation per se, but there's been a lot of discussion about ensembles of biogeochemistry, and I just wanted to bring this in as a useful demonstration. So when you run a biogeochemical model, you have a lot of parameters that you have to choose, and those parameters are very poorly known in nature. We have some lab experiments for growth rates and so on, but we really don't know them very well. So this is a Monte Carlo experiment where we did a realization of 200 members, 200 realizations of different parameters. And then we developed a cost function which was based on surface chlorophyll and deviations of surface chlorophyll offshore, surface chlorophyll nearshore, and then deviations from a climatological nutrient field, because that seemed like a very straightforward, well-constrained problem. And what I'm showing on the upper panels, like the blue, green, and red curves, is the value of the cost function for each ensemble member. And you can see that the left is stretched and the right is compressed. And what that means is that out of the 200 experiments, most of them had pretty bad cost functions, meaning they created realizations of the biogeochemistry that wasn't really consistent with the observations that we had to test them against. Whereas there were maybe 10 or 15 that were actually pretty good. And we can't really distinguish between these parameters very well. They produce very different rates. They produce very different, well, the distributions they end up producing are very similar. And so some of that is because parameters have correlated effects. But my point in showing this is that yesterday we were talking about how maybe we don't have to do a huge ensemble. Maybe we can have a limited ensemble of biogeochemical models. And I think this supports that. In our case, 10 or 20 showed some spread in behavior, but pretty similar, couldn't be distinguished very much from from the data that we had to compare them to. Finally, we found that doing a Monte Carlo simulation was very inefficient, and you could actually get to the same endpoints through more machine learning algorithms of directed search approaches to basically go downhill with these parameter changes. Okay, now I'd like to move on to the biogeochemical reanalysis. So the first thing that distinguishes the biogeochemistry from the physics is that the physical variables are Gaussian distributed, whereas the biogeochemical variables are better represented by log normal distributions. So they're skewed with a lot of low concentrations and a few high concentrations, and not normally distributed. So we had to account for that. This is a figure by Janet Campbell looking at surface chlorophyll that shows that it's reasonably Gaussian once log transformed. So we do, oh, I forgot about this. Sorry, my third slide. What's the point here? The point is simply that we do coupled physical and biogeochemical assimilation by creating an augmented state vector that includes both the physics and the biology, and we can do this log transform for part of the state vector and not for others. sorry, I'll just skip over that. This was a model twin experiment where we showed that in the model twin, we were able to get lowest biological and physical errors by doing the coupled problem. So the physics observations were informing the biology, and we found the biology estimates of surface chlorophyll were feeding back on the physics and changed the transport and changed the salinity and temperature distributions. Not by much. The physics had a bigger effect on the biology than the biology on the physics, but nonetheless, in this coupled system, everything was affecting everything. In realistic systems, we don't find as strong a relationship between biological data on the physics, but I think we're still trying to understand that better. Okay, I showed this figure because this is a demonstration of the coupled system. This is a Hofmüller diagram in those right panels where I'm showing time on the x-axis and latitude on the y-axis, and it's the chlorophyll between the coast and 100 kilometers offshore, averaged. In the upper panel is the data. This is a 1-year simulation from 2000, so it's Wijffels data, and the white represented cloud cover. In our case, the middle panel was our free-running model with no assimilation, and then the bottom panel was this coupled biogeochemical assimilation one. Now, several things I'd like to say. One is that the free-running model has some structure that's similar to the data, but it really wasn't as similar as we might have liked. Now, we could spend a lot of time improving the biological model, and we should, but no matter how much we improve it, there are always going to be discrepancies. The second thing I'd like to point out that this is the easiest metric because we have all of this data for chlorophyll. Now, we are assimilating chlorophyll and we're matching it, which is really great, but it's making changes to all of the fields like zooplankton and nutrients as well. And we don't have a lot of data to really evaluate whether those were good or bad changes. We know they're consistent with observational distributions, but they're not necessarily data constrained very well. Finally, am I late?
Got so much time here for questions too.
Okay, so in that case, I'm stuck again. I'm going to just say we really don't have enough biogeochemical data. We have fantastic things like BioArgo. Gliders collect also nutrients and oxygen and fluorescence, but we have very, very limited biological data. Much more would be very, very useful. All right, I'll go to my summary, which is that state estimation and reanalyses are a reality for physics. They are increasingly mature for biogeochemistry in some places, and I think they can be used for historical analyses and for forecasts as well.
That's great. Thanks, Chris. We have time for maybe one or two particularly incisive questions.
Hi, this is Dimitri. It's not a question, it's a comment. I think it's incredibly impressive that the cost of the physics didn't go up when you added the biogeochemistry assimilation. That is not easy. There's a lot of things that have to go right, so I'm really impressed with that.
Thank you very much. And you don't mean computational cost, because that did go up.
No, the
cost function.
Yeah, I thought that's...
Thank you.
This is Susanne Craig. Nice talk, Chris. I was wondering about the sort of error analysis you alluded to, the differences between and where you need to do the assimilation. Like if you have that spatial information, like that offset when you do have the BGC, when you do have the data comparisons, how strongly you're like needing to adjust, if there's like any spatial or temporal trends, like do you do analysis on that?
Yeah, we have areas that are consistently biased high or low, and we are consistently making changes in a particular direction in a particular season. So we do think that those are biases either in the ocean circulation, which is bringing too many nutrients or currents are too strong, or by the biogeochemical response. And so those point to ways we might improve the model, but we haven't focused on improving the model from those analyses, but that's a great point.
Matt, go ahead.
Chris, I was curious about your equally plausible utilization of your biogeochemistry in the context of parameter sets that are capable of reproducing. I think you said your objective function was the mean climatology.
No, but I apologize, I didn't say it quite right. So this was a 1-year simulation, and we had time varying data of surface chlorophyll at the surface, but we divided it into kind of an offshore and a nearshore area that we weighted equally. So we did point-by-point comparisons, but we wanted to equally weight coastal and offshore zones, so we did some magic there. But we did use climatological nutrients, subsurface profiles, because that was the only subsurface information that we had at that time. And there we were comparing to monthly climatology.
Perfect. All right, let's take a break. All right, moving on to Jake Kritzer.
Thanks, Dillon. And I want to echo Dimitri and Chris in thanking the organizers for putting this on and for the chance to speak. I also want to acknowledge and thank my 3 co-authors here, Jan, Clarissa, and Henry, my counterparts at the helms of the West Coast Regional Associations. I want to thank them both for really doing a lot of the legwork to start to put this talk together, but also for not being able to be here, because that gave me not just the opportunity to speak, but also a convenient excuse to come. So I'm going to give a fairly broad overview of the status of data collection and integration across the IU's regional associations. I want to note from the outset that, integration, in my view, is a word that means slightly different things to different people. It can mean integration of data sets into databases. It can mean integration of different types of data, integration into models, into products. I'm going to sort of loosely move around all of those definitions. So I'm going to be a little inconsistent, I think, in the use of that term. All right, so let's dig in. In the discussions that I've been part of, kind of one-on-one and breakout groups, it seems there's sort of mixed familiarity in the room with IUs. Some people, Chris and others, know us well. Others, it's a sort of nebulous entity. So I thought a quick overview of what IUS is. So the U.S. Integrated Ocean Observing System is a federal partnership among 17 different agencies, but that is run through an office within NOAA's National Ocean Service. What's unique about IUS, though, is the implementation is done through 11 regional associations. A lot of people think we're part of NOAA. We're actually not. Some are housed within universities. Others, like mine, NERACOOS, are independent 501c3s. But we are distinct from the federal government, although, again, we exist pretty much wholly and solely to implement a federal program, and that's where the majority of our funding comes from. So I put up there the mission of IUS, which is to produce, integrate, and communicate high-quality ocean, coastal, and Great Lakes information that meets the safety, economic, and stewardship needs of the nation. So a very narrow, discrete thing we're trying to do. Now, it's pretty broad in scope, and reflective of that is the fact that IUS has identified 34 different physical, chemical, and biological core variables. If there's 34 of them, it kind of questions how core they are. But I think that reflects, that this is a very broadly focused, broadly based program. To do that, the RAs all use different portfolios of observing tools. Here in the Northeast, we lean very heavily on a network of fixed moorings, but we complement that with glider deployments, with land-based high-frequency radar stations, ship-based surveys, smaller coastal stations, and so on. All those data work with a variety of different models. All of that gets made available through different data products. And all of this, importantly, is federally certified. So we sort of have the stamp of approval from the federal government that what we're doing is reasonably trustworthy. Just kind of I thought this was a useful illustration from Henry at NERACOOS and sort of how that system might look conceptually. So you can see that on the far right are some of the core variables, I don't think all of them, that NERACOOS is focusing on, and kind of how those variables are measured through a variety of different elements of the observing system, and that includes many of the observing technologies as well as different models and different types of models and maps, and then how that gets made more readily available through different types of data products, and all really to meet the needs of end users. And that's a big part of iUse, it's very end user driven. So everything we do tries to identify different types of end users, including the scientific community, the government, but also industries and recreational users of all stripes. So we're really trying to make sure that all the data collection and all the data delivery systems are actually useful to someone. This is a quick snapshot of what the state of collection of different variables that are kind of relevant to the focus of this workshop across the regions. This came from a 2020 survey. I think less than the details, what's more important is the patterns, and that is that there's a lot of effort underway to collect different types of variables that are important to ecological forecasting, but there is variability across the regions. Perhaps not surprisingly, it's the Pacific Islands and the Caribbean that are focusing more on corals. We're not doing a lot of that here in New England, not that we don't have corals. On the flip side, the Pacific Islands isn't doing a lot with HABs, because HABs are not a big issue out there. But so there's mixed portfolios, but there's certain variables and certain tools used to collect them that are deployed more uniformly across the regions. Excuse me. Now, but that table, I think, masks some important sort of trends and status of where we're at with measuring these variables. When it comes to many of the variables, I think, that are essential for ecological forecasting, so we're getting away from kind of the basic physics and getting into the BGC and some of the biological and other ecological variables, far more often than not, we're more at the research to operations stage. So iUse really aims to work at the far end of that research to operations spectrum. It's intended to be an operational oceanographic system. But with many of these variables that are not kind of the core physical variables that we've been collecting for 10 or 20 years, we're really still, more often than not at the stage of having scaled and sustained operational systems. Some are really only measured in very few regions. eDNA is only just starting to crack into the regional ring systems. HABs are, focused in some regions, but not all. Same thing with sound, imaging tools, etc. So there's a lot of kind of frontier, next generation, at least from the iUse perspective, types of observing that we're really just getting into. However, so 18 months ago, I think, end of 2020, all of the regional associations submitted new proposals to NOAA for our next 5 years of funding. And that was kind of laying out the vision for what we want to do, what we want to sustain, how we want to expand the regional ocean observing systems. In all of those proposals, these types of measurements had, I think, more prominence than ever before. So this has been really widely recognized, I think, as an underdeveloped part of the system and a pretty high priority need. And again, that's the work we want to do if fully funded. We're not fully funded. I'll talk about that a bit more in a minute. But again, I just want to reiterate that the goal is to get these measurements that are newer, at least for our systems, and that are more at the research to operations stage, to get them to the stage of being routine, sustained, and scaled operations. Oops. Why is this not moving? Did I break it? Surprisingly easy. I think this new direction was envisioned by my predecessor, who some of you might have known, Rue Morrison, who was the founding executive director and helmed the organization until his early retirement in late 2019. Rue passed away about 18 months ago. But he, I think this quote came from a bioscience article that was sort of looking at the growth in biological observing technologies, and I like this quote. So Rue at the time said, what we're seeing this explosion in how we can understand biology in the ocean. These new technologies are giving us sort of this unprecedented opportunity to understand ecosystem function. To me, that's kind of the focus of what we're talking about here, and really the next generation of what we want to do. So I'm grateful to Rue for kind of laying out that vision. Here again is what that looks like in terms of what we want to do versus what we're able to do. So Clarissa provided me this table from her renewal proposal, which shows all the different projects they've proposed. And those are the rows. The columns are the 34 different IU's core variables, so which variables factor into which projects. But the key here is the split. That blue line is the distinction between what we call our Tier 1 versus Tier 2 projects. The IU's invited proposals, 5-year proposals from the regional associations that were up to $6 million a year over 5 years. We all knew we weren't getting $6 million a year. So the idea was to create some cap space so that new funding could be brought in and add to our awards and let us do new work. But so the Tier 1 projects are, if you do nothing else, if you're at what we expect to be kind of a baseline sustained level of funding, what would you do? And then the Tier 2 projects are, as we hopefully eventually one day see budgets increase, what more would you do? It may be hard to read what's in this table. You will see that for SCOOS, there are some biologically and ecologically oriented projects at Tier 1, but many more at Tier 2. And so, if you this table kind of reflects everything below the blue line, I think, is what's going to elevate the contributions that IUs can make toward ecological forecasting, but that we're not yet able to do. So the vision is there, the intent is there, just the ability is not fully there yet. So I should have noted, we peppered throughout this presentation some of the questions, focal questions of the workshop. So I'm going to interject answers to those at periods. So one of the questions is, are these data, though, set up for easy access? And the answer is, yeah, sort of. Accessibility of data varies among regions. It also varies among data sets. The funding we have to collect data competes with the funding we need to manage data and make it available. And with some of these, at least to us, newer data sets, there's more work and more complications involved in managing and serving those data. I've said here, plankton are not equal to temperature. There's only so many ways to measure temperature. There's a whole lot of different ways to measure plankton. And trying to create data management structures that can accommodate different plankton sampling programs and then start to integrate those is a whole big can of worms. And it's an area we're not as practiced in as we are with some of the fundamental physical variables. That said, there are various data assembly centers that are in different stages of development to manage some of these emerging elements of our observing system. Sank Sound is, as you might guess, a pilot DAC that is managing acoustic data, particularly focused on the national marine sanctuaries. Clarissa yesterday talked about HabDAC, which is doing exactly what it sounds like. Out of the IU's office, the Marine Biodiversity Observation Network and Animal Telemetry Network are building systems for managing other types of biological data. FathomNet is one I'm just getting familiar with. It's kind of outside the IU's enterprise, but it's using AI and machine learning tools to help kind of understand and utilize imaging data. Within most of these DACs, there are defined data standards and data integration protocols that anyone providing data into them needs to follow. So one of the other questions was, are the regions engaged in integrating these types of variables for easy analysis and access by external users? The general answer, again, is yes. It's at different stages and looks a bit different from place to place. I'm just going to run through a few examples, but this is far from exhaustive of what's going on across the enterprise. I don't need to spend a lot of time on this one because Clarissa covered it really well yesterday, but the HABDAC is the basis for, I always want to say CHARM, but CHARM, the HAB kind of forecast mapping tool that they've developed. And that and the CHARM and the direct observations feed into this really cool HAB bulletin that SCOOS puts together that reports on marine mammal strandings and shellfish closures. And I think this is a really good example of IUS's priority of being end user driven. This is trying to help interpret and deliver outcomes of the observing system to users and in the context of various kind of economic and policy issues. Yeah, thanks. This is another one I don't need to spend a lot of time on because Sam and Emily covered this a bit yesterday, but NANUS is part of JScope, which is helping to do upwelling forecasts and a variety of complementary oceanographic forecasts that, as we heard, have value for fisheries and other uses. And in between NANUS and SCOOS, at SENCOOS, they are building sort of a framework for how they will develop and deliver biodiversity and ecosystem observations, models, and products, primarily to serve sanctuary management as well as ecosystem status reports. And I really like this. There's a lot going on here. It came out of a paper that Henry was the lead author on in oceanography recently that's worth a read. But I like how it really pulls together very distinct users, data types, and products, all kind of within the context of the food web. Have to talk a little bit about us. Forecasting has been part of what we've been doing in the Northeast for some time. A core component of our system is the Northeast Coastal Ocean Forecast System, or NECOFS, which is run about 15 miles that way, I think, by Cheng-Shen Chen and his team at SMAST at UMass Dartmouth. This is just a physical oceanographic model, so this isn't getting into ecological forecasting, but this is sort of our most significant forecasting tool. Where we're going when it comes to ecology is at present really focused on pelagic food webs. We have, the past couple of years, we've been doing new observing work with Jeff Runge at the helm to observe that little critter in the upper left. That's the lipid-rich copepod Calanus finmarchicus. We've developed a Calanus index that looks at seasonal trends through time. So the red dots you'll see on that graph to the far left are recent Calanus measurements within the context of 20 plus years of measurements. So we're seeing Calanus at distressingly low levels. That's problematic because this is arguably the most important zooplankton species in the Northwest Atlantic. It's an important prey for this little guy on the right, which is the Atlantic sand lance, an important prey species for cod as well as humpback whales. And this recent paper by Suka et al. Shows that sand lance abundance pretty closely tracks Calanus abundance. So as Calanus goes, so goes sand lance. Calanus are also the primary prey of the critically endangered North Atlantic right whale. And so a lot of our attention on Calanus these days is linked to whale conservation. These maps on the bottom are from an as yet unsubmitted paper by Camille Ross, Jeff Runge, Nick Recker, and I think a few others that is trying to use zooplankton monitoring to map the regional lipidscape or the forage base for right whales and try to predict seasonally where we may be more or less likely to encounter whales. And what we're really trying to do is, as a next step, is combine our physical oceanographic forecasts and that ecological work combine that into a more predictive way with a particular focus on Stellwagen Bank National Marine Sanctuary. So we're trying to develop a project there that has two major components. One is to better model Calanus abundance as a function of abundance we're measuring at upstream sources along the main coastal current, as well as in the deep waters of Wilkinson Basin, which is a major overwintering ground for the species. So those are two important sources of Calanus supply to the sanctuary. And then using the Calanus estimates to try to model and estimate sandlands abundance, and again, factoring in oceanography as well as top-down predation by herring. So that's where we're hoping to go. So another question we wanted to tackle was this one of whether the regions are coordinated, so among regions. I showed examples there of what individual RAs are doing, but to what extent are we coordinated among regions? Not a lot. The HABDAC is probably one good example where NERACOOS and SCOOS together are working across region and building up that DAC. And it may be something that gets expanded nationwide. The ICE Enterprise also starting to explore how the cloud can be used as a way of taking what are kind of somewhat siloed data management systems among the regions and bring them together nationally. One good example, though, where we made progress is through the Animal Telemetry Network. And this is what the portal for ATN looks like. So you can go to this map, click on any of those grid cells, and kind of see what telemetry observations of different species have been detected in that location, and it will guide you to the projects and data sets underlying that. So, and that brings together a lot of different telemetry projects that otherwise would be fairly disconnected. Limitations, though, for where we're struggling to make progress on integration, forecasting, and the kinds of things we're tackling here. I said it before, I'll probably say it many more times in the upcoming weeks, months, and years, but funding for those tier 2 activities, we need to get beyond the critical sustained systems that we want to keep running and start to expand our system. And notably, we're ready to do it. These proposals have identified the people, what the budgets are, what the plans are. So this stuff is all pretty shovel ready. We just need to be able to put it into practice. Two examples about how we're trying to move that along. Recently, under the umbrella of the IUCE Association, we produced a white paper on how IUCE is helping to track the coastal climate signal. This was really aimed at Congress and particularly the Biden administration, for which this is a rising priority, to say, we're doing this and we can do it better with more support. And that report does specify ecological forecasting as a priority need. And then some of the folks from the national office and RAs got together and put together an ocean shot for the U.S. National Committee for the Ocean Decade, which has a real focus on ecological forecasting as kind of a next generation priority for IUCE. So just to close out, how we can better work together to advance this work and meet some of these challenges. First one is answering a lot of the questions we've been talking about here, is just making sure we understand what are the priorities for variable measurement, validation, parameterization, et cetera. Again, prioritizing that and figuring out how to fund those observations, integration, and models. And then I think developing a stronger use case, which is really, I think, what's going to move decision makers. And I think what JSCOPE has done to inform fisheries management is a great example. With that, I'll wrap up.
We do have time for 1 or 2 or 3 questions, depending on how verbose the response is.
Thanks, that was a great talk. This is somewhat of a kind of esoteric, trying not to be too rabbit holey about it, but you mentioned there's a lot of observations of Calanus there. Something that the phytoplankton community are really interested in is how the restructuring of the water column and the kind of changing phenology of phytoplankton blooms affects the higher trophic levels. And I'm just wondering if there's some kind of an integration between the zooplankton and phytoplankton worlds under NERACOOS.
Not yet, but we have a proposal in review with MBON at the moment where that's a major focus. So the Calanus observing that we been doing in the last couple of years is funded by MBON. We've put in for sort of a renewal and expansion of that work, and that's one of the focus is to bring phytoplankton and zooplankton data together to see. There's a lot of focus on how Calanus drive higher trophic levels. We also want to see how primary production drives Calanus.
Yeah, and that's a signal you can see from space.
Yes.
Watch this space.
And that's, the proposal is very aware of that.
Super, thank you.
Thank you so much for this really great presentation. So I would like to go back to the integration aspect. I think all the types that you have talked about really sound really excellent. So in terms of integration across regions, so did I understand correctly that the biggest limitation is funding? So there is interest for the different IUs to join effort and maybe combine the
funding is certainly one, because we need to create the new data management structures and then get data into them. Inertia is another one, though. We have systems that have been run by the same people, producing data in the same ways and managing it the same way for a long time, and it's kind of easy to keep doing what we've been doing. So it's both just the costs, I think, are the biggest barrier, but then just the will to start managing data in a less siloed way.
What would it take to remove this inertia?
Well, I mean, the funding, but I think also just, frankly, the mandate, and the mandate can either come from the national office to the regions or from the regions to our operators. If we say to them, your funding is now going to be contingent upon, and we'll pay for it, but reshaping your data management to go into this national DAC as opposed to whatever server you have there. I mean, we need to give that directive, but we need to resource it as well.
Awesome. Let's thank Jake one more time. All right, next up is Susan Wijffels, talking more about observational networks.
Just to advance the slide, I just use that one. Just the arrows, okay. Good afternoon, everyone. So I do feel a bit like a fish out of water here today. I'm a physical oceanographer, and I mostly think about global ocean changes and the general circulation of the ocean. But I have spent a lot of my working life also worrying about the global ocean observing system, but really again from a climate focus, very large-scale focus. So towards the end of this talk, I'm going to swim right out of my lane, and I probably will make a lot of errors, so forgive me for that. And this is a very personal view after listening to today. I am starting to be involved in some BGC monitoring. I'm on a huge learning curve. And so this, again, is just a very personal view. So hopefully it'll at least get some discussion going. So I would say that around ocean observing, we are going through a kind of revolutionary period. So for what I call the blue water, the global deep ocean, we've seen the implementation of the global Argo array. And just before I continue with that, put your hand up if you don't know what Argo is. Everyone knows, good. So that is a program that's involved over 20 countries, a big international cooperation, and just uses really stupid, simple robots to go up and down in the ocean, passive, don't require any piloting, and they operate for a very long time. But the things about Argo that have worked really, really well, and I'll touch on this later, super strong data management system. We spend 30% of our budget on that. I'll get back to that. Very clear design goals, like super clear. And the design was actually informed both by satellite altimetry and previous things that have been done in WHOS for the XBT lines and so forth. So Argo's mission really is to kind of map in real time what I think of as the slow manifold of ocean variability. So this is the 1,000 kilometer seasonal 3 to 4 month change in temperature and salinity. That's what Argo can resolve. Eddies are noise to Argo. We have to average through the eddies. We attempt to do that. But now that we've had the array running and it kind of got up to global coverage around in 2004, we can map the temperature perturbation globally averaged through the deep ocean month by month in real time. This gets updated just in real time. And so you can see from the top to the bottom there, this has got the seasonal cycle taken out, obviously. There's our global warming signal. Sticks out like crazy, it goes to the sea floor. And then you can see, the back and forth of ENSO at the surface. And as ENSO warms and cools at the surface, you see the subsurface does the opposite. That largely cancels out. So if you integrate that heat content, it's just a staircase. There's no hiatus or anything like that. You can see how the tropical Pacific drags around the global average. You integrate through it. We have a staircase of warming. We need this for pH. We need this for oxygen, right? We need to go beyond the physics now and be able to track these things. The other thing I'll say is that even though a lot of the discussions here are very regional, I feel very strongly you have to measure globally to really understand regional changes and to attribute them. And so we need this global backbone, I believe. And so, we have this original core version of Argo, and it's in a context of a bigger observing system. But I think this idea of a backbone is a very useful concept too for regional observing systems. Let's see if it'll let me do this. All right. Now, the other really amazing, wonderful tools that our community now has is the constellation of Earth observing satellites. And these are, again, absolutely critical, vital infrastructure that we all rely on. And I know there's a talk next about these. But these are, again, absolutely essential. And we should all tell all our program managers how much we love them and need them, because we do. And so I've just listed some of the key parameters up there. We have to keep those missions going. But the other reason I wanted to bring this slide up was that as we build these systems, we have to be building them in a kind of integrated design way. We have to understand the synergies and interdependence of these different networks. And I'll just mention a few. For instance, the surface topography missions, which map the eddies in the ocean, actually help you de-eddy Argo. So if you're trying to map the slow manifold from Argo, you can actually use the altimeter to take out some of the altimetric noise and reveal the slow manifold much more clearly. And in fact, we also use Argo data to help us just understand when sensors go bad. If a sensor goes bad, Argo-derived sea level departs from the altimetry, and we say we've got a problem with a pressure sensor or a salinity sensor or something. So there's all these interdependencies. Satellite sea surface temperature, absolutely essential for so many applications, very reliant on the surface drifter network for CalVal. Surface drifted technology is ancient, very old, very simple, and yet absolutely essential to CalVal SST. So there are all these interdependence, and I think as we design our observing systems, we should keep the full system in our minds, the satellite system and the in situ system, and how they combine to support each other to produce the information we want. I really think we should be thinking about it in that way. So I was curious when I was asked to give this talk about the Northeast. This is about the Northeast area. What do the data flows look like? Okay, and if I was someone wanting to build a reanalysis, I'd go to the World Ocean Database. That's where all the data is meant to end up, and ask myself, okay, what does the TS sampling look like? So I chose 2018 and 2019. That gives enough time for some of the data, the delays that we know in the data system getting back to the archives. And so that's 2 years of monthly data. So, every 2 months is plotted there. And so you can see in magenta, the Argo system offshore. And on the shelf, you can see some ship-based stuff in cyan and the odd glider track. But what really struck me is that there are months where over a 2-year period, huge parts of the shelf have almost no sampling. Now, it could be that there was no sampling, or there could be that the data just hasn't flown to this archive. And yet this is the archive where a reanalysis team is going to go and build a global reanalysis or get data to check what's going on. And so, this struck me as a problem in that the offshore looks better sampled in months than the shelf. That's a big problem because the shelf has like really fast timescales and really short space scales. So that puzzled me and worries me. Okay. Now let's look at oxygen. So this is what you get for oxygen back in 2018 and 19. Now maybe there's much more oxygen being measured, but it's not at NCEI. It's not at the World Ocean Database. So this is terrible. And so I think that, this is kind of laying out to me starkly what the challenge is. And we've heard over the last 2 days how subsurface profiles, and particularly the biogeochemical challenge, how hungry our systems are. We have these incredibly constructed, capable modeling and data assimilation systems, but we just don't have anything to feed them with. So this is a big challenge, I think, that needs to be focused on. So I'll just dig into this a little more later on, but I'll just say, back in 2019, when a lot of people who think about ocean observing met on the Lido in Venice, and we were thinking about the next big challenges. At that point, at the end of the meeting, we said, well, it's to go multidisciplinary, it's to go towards the coast, and it's to go deep. And I would say in the intervening 20 years, we've made some progress, but less than you would like. But I'll just say that a lot of background work's being done, and I think we're ready to go now. There's a lot of capability building that's happened. So I would say for offshore, sustained and deliberate, like really broad-scale BTC measurements are just beginning. They're just on the verge of being a reality, and I'll show you a bit about that. On the shelves, I worry we're going backwards. I tried to get the plot done before the meeting, but I couldn't quite make it. But in Australia, this is certainly true. In the 50s and 60s, there were lots of regional research vessels doing shelf surveys all the time, including oxygen and nutrients. Many of those have been thinned out to almost nothing, and we haven't put in modern technologies to replace them. So on the Australian shelf, the oxygen coverage actually back in the 50s and 60s was quite good, but it's not anymore because we haven't replaced those observing arrays. So we're kind of gone backwards in some places, and I don't know if that's true here, but it could be. The offshore sampling, I feel, has been really enabled by platform maturity. So here I'm talking about the profiling float platform and glider technology, and for the surface, we might be seeing some surface AUVs come in there. But the other big critical ingredient here is sensor maturity. And so, those low power, low mass, and very stable BGC and optical sensors are mostly here. Some of them are still problematic, but they're worthy of now starting to deploy at large numbers. We have, I'm going to show you this a bit in a minute, we do have a global observing design, at least for a global float biogeochemical array, unless I find it difficult to find one for glider sampling on shelves. I haven't found that yet, and I wonder if that's a target or something that should be thought about. So this is a new design for the global Argo array. So the original Argo array just measured temperature and salinity just to 2,000 meters. It didn't operate in the polar zones, didn't largely operate in marginal seas. So this design here has us moving into seasonal sea ice zones, and it converts basically 1,200 of the floats to deep reaching floats, and it converts about 1,000 of them to biogeochemical measuring floats. And the biogeochemical measuring floats are ultimately going to try to have 6 parameters: nitrate, oxygen, pH, chlorophyll, and CDOM particulates and irradiance. And so we're in a situation now where we're just building capacity to do it. and are hoping we can launch, if we get support, into implementation. So this is the current status of the global biogeochemical Argo array. And here I've just taken the subset of floats that are measuring pH, because the pH floats are close to having those 5 parameters on them. And so it's just really been a series of regional pilots that are starting to build out now into a global network. In the US here, we've had really wonderful support from the National Science Foundation with 2 huge kind of arrays, the Sulagna Ray, which accounts for most of those Southern Ocean floats you see there, and now the GoBDC array, which is going to put 500 of these out in the global ocean. But other nations are really, really struggling to match that. And part of the reason is that we still are facing really high sensor prices and low supply. And what's happened with COVID is not helping. And then the other major reason is just a scale up in funds. And so just as we heard with Argus, to operate the full one Argo design, we believe we have to triple the operational cost globally for Argo. So it's going to go from a 35 million a year program globally to a 100 million a year program, which is a bargain, because a satellite sensor costs about $100 million a year, a launch satellite, right? So the power of what we can do with this is huge, but we just have to sort of start scaling up the investment to match what we invest in satellite sensors. And I would argue it's easily a good deal. But just again, to drive home, this is a nascent network and it's not secure, and we need community championing to make it happen. So it's not in the bag by any imagination. And in fact, if we don't start getting serious money to support this, we might have to just go back and stick to our knitting and operate the core array, because this is going to basically degrade the core array if we can't get the right support in place here. The other really, really important part that's made Argo super successful is our data system. And I know we've heard a lot about that, but it was groundbreaking when we started this 20 years ago. And the thing our goal here was to put a data system in place that would feed operational forecast systems. So we have a very well-defined real-time data flow with very well-defined metrics of timeliness. We're almost down now to 6-hour delivery. We want to get it down to 3-hour delivery for hurricane forecasting. And that's all tracked. But the other part is that we understand Argo data are also going to be used for climate change detection and detecting very tiny signals. So that's why we spend an obscene amount of money on our sensors, and we also spend a huge amount of money on climate kind of very high level of quality control. We don't do perfect, but we do very well. And so here we have a database where data gets out very quickly, and then a year later, every profile gets replaced mostly with a very highly flagged and QC'd and adjusted profile for climate reanalysis. And so that's made the user database of this data set very, very large. And then there's a single source of the data. You can go to the GDAC and download the global data set, or it's all at NCEI and some of the other global repositories. So the data system really has been essential. And again, I think we invest between 20 and 30% of the cost of running the network on our data system, and every cent is well spent, I would say. Amazing team. So I'll just talk a bit about what I see on the slopes and shelves. And so this is a really lovely figure from Robert Todd's Ocean Obs paper, just showing some of the glider observations, the routine glider observations happening along the east coast of the US and the west coast. And so these are the kind of deliberate repeat observations where you can start really building up time series from the glider data sets. But you can see that East Coast gliders are largely in the blue water still. It's a Gulf Stream focused thing. There are shelf glider observations, but as you saw from my previous look at what's in the archives, is the coverage is very intermittent and doesn't look like it's actually following some kind of space-time sample design at all. So, I think there's an opportunity here to really do something really interesting with the gliders. The reason I'm talking about the gliders is that they can carry a lot of the same biogeochemical sensors that Argo is being equipped with. And so if we do it right, you'll just have a seamless parameter set from offshore to onshore if we do the data management correctly and work together. And, we are starting to do that. So I guess one of the things that struck me thinking and looking at these plots and what's been done to date is, can there be a clearer kind of design process for glider sampling on the shelves? And here I mean really use some of the incredible modeling systems and data assimilation systems that we've heard about to really inform where the gliders should be sampling and how frequently they should be sampling. So that you can really understand for the whole East Coast shelf, the whole West Coast shelf, where the coverage needs to be. Another big question is what BGC and optical parameters should be carried. And again, really make sure that the data management is seamless between Argo and the glider data sets. The platforms are a little different. Gliders move through the water faster. They have errors, much bigger sensor dynamic error, but we should be able to make them really seamless. And again, we should be just demanding this now. This is 2022. We should demand that everybody uses common standards, agreed formats, agreed ontologies, and the data should all be out there in real time. You can share oxygen data on the GTS through Buffer. Argo's got a proposal in to share all the other biogeochemical sensors. That should just be happening par for course. And I will say I just couldn't find a single place to go download the glider data. That's a problem. That should not happen. If I was a reanalysis group, I would probably go crazy having to go around, refresh, update. That's not good enough. We have to make it easy for our friends to use our data. So the future challenges in my mind remains that we have some big sensor challenges, so we need to find some funding to push this along. Super high prices for some of our sensors. We have dynamic errors in oxygen, pH, all those things. So you push one of those sensors through a strong temperature gradient, their accuracy plummets, so we have to deal with that. And we need to find ways to intercalibrate optical sensors. So again, it doesn't matter what platform the sensor is on, you can compare one to the other. I think, in most developed nations, access to ship time is going down and down and down and down. It's just a reality. It seems to be particularly for blue water platforms, but possibly on the shelf too. So we're just going to have to rely more and more on these autonomous platforms. So we need to drive up longevity, power efficiency, and their reliability. And we need to build capacity across more teams to operate them. And just a small point about the cost of communications, which hopefully there's good news on the horizon. This is where I'm really swimming out of my lane here. I think, in my small, more recent experience of trying to understand how you do biological observations at scale, the fact that we might not be able to catch water very frequently means that we probably have to go to sensors that can operate on their own. And the things that got me very excited, probably out of ignorance, biooptical imaging and bioacoustics. I think there's going to be a huge amount that could be done with those. And so I think there's probably a sense of urgency around having to start piloting them, deploying them on different platforms, and then build really modern, state-of-the-art data management systems for them. And I would say immediately just go global. Talk to your international partners and figure out how to analyze this data, produce products, and produce a globally understood data set from some of these new technologies. I think it's a super cool opportunity right now. And then with eDNA, it does presently require catching water, but I think there are systems being designed now where you might not have to do that. And so I think that's very exciting. And, there are many people that have written papers about this. This is one again from the Oceanic set from Frontiers, saying just how this combination of eDNA and optoacoustic could be very, very powerful. So I'll just finish up and say I think we've made a lot of progress, particularly in the blue water. And I would just want to stress this really important idea to remember the synergy that we have between the satellite observing constellation and the in situ systems. They have to nourish each other, and we need to build them to do that. Scaling up BGC coverage offshore, I think, is a major, well, offshore and on the shelf, major immediate challenge. I think we can go far if we just given a lot of money, we've done a lot of the groundwork, we're ready to go, but we also have to deal with sensor prices and availability. My view of the coverage of the sea, just the basic physics on the shelves, it looks really uneven to me, and I just wonder whether a really good design project around glider sampling with a national view could really benefit the community, and put something in front of funders that really feeds forecasting, because they can produce real-time data sets across many different parameters, and it could be a really good story. And to think about whether there should be a backbone minimal sampling using gliders or some equivalent platform. I've already talked about data distribution and management, and comment on the optical imaging. So.
Probably just have time for one pressing question.
Oh,
sorry.
All right. Yeah, hi, this is Dimitris Menemenlis. Quick question. I'm wondering if part of the cost for Argo BGC is because they have to go to 2-kilometer depth, and whether you could save some costs, and whether you need to go to 2-kilometer depth for BGC variables, and if not, whether some costs could be saved there. Just curiosity.
I think you do. I think you do for oxygen. I think you do for particulates to get the biological rain out. So yes, I think you do. What's built into that one Argo design is a one-for-one swap, right? Like a BGC float is replacing a core float. It's on exactly the same mission. And so basically partly the platform's for free, right? So, that's the most efficient array we could build. We could have a completely separate BGC array and just stick to our knitting on core, but it's much more powerful and more efficient money-wise to integrate them.
So we have one online question, so we should at least let that be heard. It's from Mark Oman. I don't think most ecological variables, especially measurements of organisms, have an open ocean bias. It's really the opposite. For nearly all consensus of organisms, the coastal ocean is disproportionately represented. Thank you.
All right, let's thank Susan one more time. All right, and we will, if you have more questions, please keep them in mind. We'll take them at the end. Next is Susanne Craig, who will take us home before lightning talks. That's her.
Good afternoon, everyone. Just a quick thanks to the organising committee for inviting me to my first in-person event in 2 whole years. I think the last time I saw real life scientists was in San Diego at the last Ocean Sciences meeting. Okay, I want to tell you a little bit of a story today, and that is about using ocean colour in the coastal zone. Why you should do it and why you might want to be careful how you do it. So just a kind of brief statement, get us all on the same page. So ocean colour and things that we call inherent optical properties, or IOPs for short, are very robust for the most part proxies of biogeochemical processes in the ocean. We can quite accurately estimate them from satellite ocean colour in many regions of the global ocean, using approaches such as the tried and tested band ratio models and a whole array of semi-analytical approaches. However, what we see rather frequently is that we're not able to quantitatively use that satellite ocean colour data in a variety of scenarios, and I've listed some examples there. Those may be in waters or conditions under which some of the model assumptions, the ocean colour model assumptions, are violated. And this second point here, which is what I'm going to talk quite a bit about today, is the difficulty, the perennial difficulty that we have in removing the atmospheric contribution to the signal detected by the satellite. And unfortunately, that is typically over coastal and inland waters, the very waters that we are talking about this week. So we really have to push for alternative approaches if we're to fully exploit the power of ocean color remote sensing. So I just want to show you some cartoons, some brief cartoons here. What the satellite sees, of course, is a summation of contributions of light scattered by the atmosphere and the light exiting the air-sea interface. So unfortunately for us, where we are really concerned about this water-leaving component, that is what we want and what all of our models are based on, that water-leaving component only comprises about 10% at the very most of what the satellite actually sees. So what we have to do is remove, in a mathematical sense, the effects of the atmosphere. So how do we do that? I'm not going to go through all this. It's an equation. Just look at the pretty color boxes. Okay. It's late. We're tired. What we want is this thing here, LT, top of the atmosphere, as the satellite sees. Use my cursor here. That's what we want to get to. And this whole kind of mess in the middle, this smorgasbord of various radiative transfer terms, is what we want to subtract. And we do that using a combination of measurements, models, and assumptions. I also want to introduce this concept of what we call optical complexity of the water body, where our blue open ocean waters are what we would class as fairly optically simple. And what do I mean by that? I mean that the colour of the ocean is dominated by the presence of phytoplankton. And all other optically active constituents, so things like suspended sediments, colour dissolved organic matter, they all move along with the chlorophyll levels in a commensurate way. As we transition from the open ocean basins into the shelf seas and right into our inland and coastal waters, we enter into what we class as optically complex waters. And that is a scenario in which those other optically active water constituents other than phytoplankton, they move independently of each other, which is pretty problematic for lots of reasons. I can tell you all about them over a beer, but it's problematic. And the take-home message from this little slide here is that this atmospheric correction becomes increasingly challenging as we move into the coastal waters and inland waters. Here's just an example of how wrong it can go. I'm showing you a whole bunch of remote sensing reflectance spectra. Oh, font problem. Okay, sorry, font problem. So the spectra that are in the heavy black lines are atmospherically corrected spectra where it's gone a bit wrong. We have subtracted too much atmosphere and we end up with negative reflectances. And the thing to note is that that effect happens most strongly in the blue part of the spectrum. Okay, so just an example, if you were to try to pull up some chlorophyll imagery, so chlorophyll being one of the Ocean Color Data products, operational products, you would see something like this with lots of missing pixels. Or around the coastlines, they would just be flagged, and they're flagged because of atmospheric correction and failure or adjacency effect, which just means that the land is bright, the water is dark, and sometimes the brightness of the pixel seeps over, so they get flagged. And rather inconveniently for us as coastal oceanographers, that's where all the action is happening. So what do we do about it? This approach came to me after an extended, frustrating period when I was running a coastal observation system where I could not get any ocean color algorithm to reliably retrieve any parameters of interest. So I threw all of the semi-analytical radiative transfer stuff away, looked at my statistician colleague and said, well, I do it. And he said, why are you trying all these like fancy algorithms? Just try an EOF analysis. So what I'm showing you here in a little cartoon is the kind of evolution of an idea that myself my colleague Chris Jones came up with a number of years ago now, which is just simply to take do an EOF analysis of the top of the atmosphere spectra. Don't bother with that fancy atmospheric correction routine. Just use the EOF analysis to try to tease out the underlying variance in that signal that are due to the water constituents themselves. So we tested that idea using ocean color algorithm test data set. This is the benchmark data set, highly QC, QA'd test data set that all of NASA's ocean color algorithms are trained and developed on. It's called NOMAD, publicly available at that website. You can see it's heavily biased towards the coastal regions, which is where we do a lot of our oceanography. I don't want to bore you with this really busy slide. Suffice to say, this is the result of running that top of atmosphere EOF-based algorithm, where we've built models for chlorophyll and several inherent optical properties, which, as I told you, act as really robust biogeochemical proxies for certain processes. I'm zooming in on one of them, and that's the phytoplankton absorption coefficient, sexual absorption coefficient, because it's a rather nice alternative phytoplankton biomass proxy. You see there that in the model you get nice homoscedastic distribution of the points around the one-to-one line. We do some tests for overfitting, do a bit of bootstrapping, cross-validation, suggesting that the approach is pretty robust and likely not overtrained. I looked at this a little bit further by doing a sensitivity analysis using a coupled ocean atmosphere model, where I played with what was in the water, how much stuff was in the water, how thick the overlying layer of atmosphere was, so the aerosol optical depth. And it turns out that for all of those different scenarios, for all of the different wavelengths that I tested it at, that the model really does a rather good job. So take-home message is, hey, maybe we don't have to struggle with this pesky atmospheric correction after all. So that was the genesis of this kind of idea that I've gone on to develop with my data scientist colleague, Carlo, a former NASA colleague. He's now moved on to industry, but we still collaborate. This paper is in the preprint Ecosystem and Earth Archive. You can find it at the Earth Archive repository there if you're interested in it. So what did we do? We took that basic concept of not bothering with atmospheric correction, but we translated it into a Bayesian machine learning approach. So we worked in a Bayesian framework. Why did we do that? Just some of our underlying reasoning. We all know we're chronically undersampling the ocean. At least for what I'm interested in, I have a lot of sparse data, which leads to a lot of overfitted, non-generalizable models. This particular approach that we converged on intrinsically mitigates overfitting just by its very nature. It's a principled modelling approach and allows the inclusion of prior knowledge, meaning that it requires a very close collaboration between us as the domain specialists, so oceanographers of various flavours, and the data scientists. So it's not just, hey, Dr. Data Scientist, here's a whole bunch of data, have at it. It's a very close collaboration, a true collaboration. And it really embraces what the human practitioner knows about the physical system in question. And of course, because it's Bayesian, we can integrate measurement uncertainty from all of the things that we measure. It's readily adaptable to clustering data, the clustering in the data sets that often occur just in nature. And as we collect more and more data, as often happens in ocean color, we can constantly update that model and improve our priors. I'm sorry about these fonts. I think I had Calibri installed in my machine and it's not native to the Mac, so sorry about the fonts. So what did we do? We built this Bayesian neural network, and we chose to go the neural network route. Let's just gloss over the details. The take-home message being that we used that same approach of taking EOS at the top of the atmosphere signal. We did not try to atmospherically correct, which means that we can work further into those coastal regions without heaping up lots of errors due to poor atmospheric correction. We put in different model features such as latitude, longitudes, day of year, etc., etc. But it turns out that the optical features are actually the strongest predictors, unsurprisingly. So this is a kind of way to visualize the data that I really have grown to dislike, the scatter plot, but it's a nice kind of way to do a quick and dirty visual representation. What I'm showing is what we actually measured versus what our Bayesian neural network predicted. note that the points are actually the means of a predicted distribution, because that's what we get with the Bayesian approach. And this, other points to note, this is out of sample observed versus prediction mean. Okay, so we get really quite nice error metrics there. Okay, how does that compare with off-the-shelf operational algorithms? What I'm showing here is how the Bayesian neural network compares to the GIOP model, which is a NASA operational data product that does phytoplankton absorption. Our Bayesian neural net is in the circles. The off-the-shelf product is in the triangles, and we see that we do really rather well using the Bayesian approach, especially at the blue wavelengths. And remember, that is where we have a lot of difficulty in achieving atmospheric correction. Okay, so take-home messages. This type of machine learning approach was able to accurately estimate an optical biomass proxy for phytoplankton, AFE, and chlorophyll. I didn't show it for brevity here, from top of the atmosphere spectra. That means that the standard atmospheric correction procedure was not required, and that's particularly relevant for the very waters that we're speaking about today, these optically complex coastal waters. And of course, being that Bayesian approach, it gives us robust estimates of uncertainty, resistant to overfitting, and improves as we gather more labeled data. Okay, so let me stay on this Bayes train and tell you a little bit more of the story. So if you're ever looking for a light read on a plane journey, check out this book by Sharon Grain. It's all about the history of Bayes, the theory that would not die. Anyway, just a little factoid. So we recently, myself and this team of wonderful humans here, received some funding from NASA to undertake machine learning approaches for predicting phytoplankton community composition from ocean colour. So we are a team of data scientists, so Carlo and even Carol are data scientists, and then wonderful Jason Graff and Susanne Craig, and our biogeochemical modeler is Cecile Rousseau, also at NASA with me. Okay, so what are we doing, and why is it relevant to you? Again, phone problems, sorry. We are, the problem that we wanted to address, and I'm hoping this is of some relevance to those of you who think about phytoplankton groups and how to represent them in your models. Traditionally what's happened is we've developed models that that derive an empirical relationship between HPLC diagnostic pigments that are within the phytoplankton themselves and ocean colour. And these are very empirically derived models. However, it turns out that pigments are a really very imperfect proxy for PCC, or phytoplankton community composition. They can co-occur across very different taxonomic groups and sizes and functions. And it turns out that with environmental and physiological forcing, those diagnostic pigments actually are very plastic, they're very dynamic, and they can alter their relative abundances, which means that as we experience more and more climate forcing, we're looking at restructuring of water column, which in turn perturbs physiology, and the pigments and the relationships derived empirically are no longer valid. That is not to dismiss the last 40 or 50 years where people have been trying these approaches. It was all we had. HPLC was what we did for the longest time, and there is 50, 60 plus, 40 to 50 years worth of HPLC data now in world ocean databases. So here are the ingredients. We're using machine learning, and what I'm going to refer to as less ambiguous metrics of PCC. And those include something we've heard a lot about this week, the imaging flow cytobuoys, IFCB, traditional flow cytometry, good old microscopic enumeration, and even some molecular approaches. We're going to use the top of atmosphere approach again, which I told you about in the previous slides. We're going to look at contextual environmental information, which comes for free if you're doing satellite observations, importantly, along with some kind of associated measurement uncertainties from those measurement platforms that we intend to use. And then this kind of vague term on the end, Google it, it's a thing, your known and your unknown unknowns, which can be represented fairly robustly in a kind of way that is not nonsensical. So those are the ingredients of our model. What are we doing right now? We are trying to figure out exactly what metric of phytoplankton community composition we should be using. We can measure it lots of different ways. As someone alluded to in a couple of talks ago, you can measure plankton in a gazillion different ways. So which one should we use as our predicted variable? So unknown, and we were pretty upfront about that when we proposed this idea. So what we're doing right now is we are doing exploratory data analysis of a synthetic data set using the NERACOOS model out of the NASA GMEO office. There's a coupled ocean atmosphere radiative transfer model with a full biogeochemical model in there as well, and that is run by Cecile, so it represents 6 phytoplankton groups. So what are we doing right now? We can detect the optical signatures. I think I've convinced you that we can do that pretty easily. We want to codify ecological principles in Bayesian models and conduct a sensitivity analysis that's going to tell us something about what we might be able to do in a real life scenario where we have a limited set of model features that we measure. So that was my little story about why you should use ocean color. I want to finish off by just telling you quite briefly about what NASA has in terms of satellite assets. You're all familiar with CZCS, MODIS, Aqua and Terra, MERIS, now deceased, assets in the International Space Centre, etc. But what's upcoming to take over the mantle of MODIS, and that's the PACE mission. That's the Plankton Aerosol Cloud and Ocean Ecosystem mission. Yes, play good. So this is a hyperspectral mission. PACE will be launched with our COVID delays factored in around January of 2024. It consists of 2 flavors of instruments, the ocean color instrument, but also 2 polarimetric instruments, which are primarily intended to characterize the atmosphere, but can also be used for ocean studies with a bit of creative data analysis. Okay, so it'll be launched, as I said, January 2024 from Cape Canaveral on a SpaceX Falcon 9. Those are just a list of the spectral characteristics. It's hyperspectral, and then with some discrete and broad bands in the near IR and shortwave infrared bands. And it will have approximately a 1 kilometre square footprint at nadir. Just to show you a kind of one of these thermal diagrams again, this is the kind of time and length scale that PACE will be able to cover. So it lies right in there in the middle. I've drawn lines through since deceased proposed projects there. Another upcoming mission that some of you may be aware of is GLIVAR, short for the Geosynchronous Literal Imaging and Monitoring Radiometer, and this is particularly relevant for the coastal image, the coastal waters of the US. It's America's first geostationary satellite, funded under NASA's EVI portfolio. PI is Drew Salisbury at UNH. And here are the areas of interest. So rejoice ye all. The Northeast is very well represented, as well as the Gulf of Mexico and some other key areas, with targeting also for key hab sites. Okay, so what will GLIVAR do? It is also a hyperspectral mission. There are its characteristics. High temporal resolution, so we can really get at process studies. These things will be imaged up to 6 times a day, depending on which region of interest you're in. And of course, the idea is that here are the length and time scales that GLIVAR will cover, and the idea being that we can really resolve these highly dynamic coastal features. So watch out for GLIVAR. We'll be doing a lot more posting in that. Okay, just a couple of last thoughts to finish off. As well as satellite assets, we also have our suborbital assets that we've heard quite a lot of them from the integrated ocean observing systems. UAVs are something we haven't sort of spoken about. It's a particular pet project of mine. I currently have some funding to do this. UAVs and unmanned aerial vehicles, low cost, drones with hyperspectral ocean color sensors on them, as well as other types of payloads, and they cover these very small length and spatial scales that we're interested in, and deployed in a fleeted fashion could really cover a lot of the gaps left by satellites. Okay, mooring ships, opportunity gliders, wave gliders, sail drones are some of the other assets that can really fill in those gaps left by satellite. Okay, I just want to finish off by addressing specifically some of the questions that Victoria primed us with. In terms of ensuring data fidelity and repository and all of that good stuff that we've been talking about, all NASA-funded investigators they're contractually obliged to submit their data to our repository at CBAS, Ocean Color, plus all of the in-situ data that goes along with it. My own personal recommendation is something that hear a lot increasingly spoken about is this idea of adhering as a community to FAIR data principles, findable, accessible, interoperable, reusable. You can check out gofair.org for those. It does require funded support to achieve this. So this is tell your program managers, this doesn't happen by magic. It requires data scientists and people that know what they're doing to build these systems. NASA is also at agency level encouraging a push to open science. You can check out the transfer to open science philosophy at that page there. And then just to finish off, specifically for the PACE mission, something that we've had a lot of success with, and I think Clarissa is actually, she was telling me she's been roped into this. The PACE mission has had a dedicated applications team since its inception. That's run by Erin Urquhart and Natasha Sadoff. That also includes the development of an early adopters program where we're priming a future community of stakeholders and data users. And along with that goes a community of practice that folks can sign up for. That's a free thing to do. So you can check that out on our PACE website, reach out to me or Natasha or Erin. And just because we can, and I had this, here's some time lapse of the PACE Ocean Color instrument being prepared for thermal vac testing. I'll let that run in the background as we wrap up. It's kind of cool. Thank you for your time.
We've got time for one or two questions. I'll ask one since we don't talk about that. I'll ask one anyway.
Okay.
10 years ago at a satellite conference, I saw a couple of posters of people doing speciation of phytoplankton from satellites. I've never really seen any papers since that time. Is that something that's commonly done now?
Yeah, great question. Okay, so that what I alluded to in those first few slides, talking about the phytoplankton community composition is exactly what you're talking about, where we use those empirically derived, rather imperfect proxies for who's in the water and who's doing what. So big things like diatoms and little things like Prochlorococcus or Synechococcus. We've been doing that using those pigments for about 20 to 30 years, rather imperfectly and rather frustratingly. And the community, if you ever see me get angry at a conference, someone has said phytoplankton functional type from Ocean Color, and my brain exploded. It's a phytoplankton community composition because it can't do function yet from space. But yes, that is a big challenge for us, and it's exactly what we're funded to do. Yeah.
One more question from Charlie.
Sure, there's a book by. You mentioned the NOMAD database, which is my understanding is used to calibrate all the operational. This always struck me as very limited in its spatial coverage. Why is that?
So, NOMAD is a subset of all of the data. So let me preface this. NOMAD version 1 was published back in, Sean Bailey and Jeremy Wardell published that paper back when they were young saplings in, I think, 2012. And it has not been updated since then. So it was a subset of all of the optical data that was available, along with labeled data validation points of the entire CBAS repository. You saw from the distribution is very coastally biased, which is great for me because that's what I'm interested in. We now have someone, Violeta Calzado, is now taken over NOMAD, and she's working on a NOMAD version 2 that will be hyperspectral to support PACE, but also just more voluminous and hopefully better resolution. But the problem, as always, is getting these coincident measurements of ocean colour plus what's in the water at the time of the optical measurement, and that becomes the limiting factor. Yeah, so that is why we have small n's for these benchmark data sets.
Yep. Okay, well, let's thank all 5 of our afternoon speakers.
All right, so I think we're going to take a 15-minute break. We'll meet back here at the 25 mark for the poster spotlight talks.
I think you're able to hear me well.
Yep.
Thanks. My name is Sulagna Ray, and I'm a support scientist at Environmental Modeling Center, working on subseasonal to seasonal forecasting development towards NOAA's next generation forecasting system. However, today I'm going to present my study that I have continued from my affiliation with University of Connecticut. I'd like to thank my collaborators listed here. And we recently submitted this study. This is a follow-up study from our earlier published paper looking at the drivers of the subsurface temperature variability in the Northern California current system, looking at the North Pacific and the tropical Pacific drivers. In this paper, we are focusing on the winter subsurface temperatures at depth. And this figure, Samantha already has introduced to you yesterday. We are looking at the using the CFSR reanalysis data for the period of 1979 to 2017. We try to do a heat budget analysis of the green boxed domain here shown in the left panel. So these are the correlations between the temperatures at depth averaged within the black box region, which is defined as the NCCS domain, to in the summer to the winter prior conditions around the region. And you can clearly see that during the ENSO neutral years, we have a stronger correlation. So what this study shows is demonstrates the capability of using a heat budget in the intermediate layer below the mixed layer in estimating the what are the dominant processes that's driving the temperatures at depth in this domain. This framework was built during my affiliation with GFDL in collaboration with Andrew Wittenberg and Stephen Griffiths, and I use the same heat budget in a slightly different way in this study. Can we go to the next slide, please? So here it is, the upper left panel is showing the intermediate layer where the upper mixed layer is the topmost layer, the schematic is showing, and we have a deeper layer which whose bottom boundary is an isopycnal layer. We estimate the heat budget in these two layers and estimate the intermediate heat budget as a difference between these two layers. So focusing on the, not going into too much details of it, the right-hand panel shows the evolution of the heat budget terms within these ESN domain, and focusing on the circled region, you can clearly see that during the winter months, you have a much thinner intermediate layer with the deepening of the mixed layer. The advection, which is the green bar, is the most dominant driver of the intermediate layer total heating, which is the dark orange bars. Now, if you decompose the advection term, you would find the lower panel figure in which the decrease in meridional advective cooling in these domain facilitates an increase in the total advective heating of this domain. And this is along with net zonal advective heating, but that is in a direction opposing to the increase in the total advective heating. Now, this is also the time of the year where you have a seasonal appearance of the poleward flowing CUC, the California undercurrent, as you can see in the contoured plots in the lower left panel. So overall, it's likely that the CUC plays a leading role in preconditioning the winter subsurface for the following summer upwelled water. Can we go to the next slide, please? So here I'm demonstrating what happens in the heat budget in an El Nino composite, and focusing again on the arrow region. You can see an increase in the total heating of the anomalous heating of the intermediate layer, and the advective heating again dominates total heating of the intermediate layer. And the components, the panel below is showing that all the components contributes towards the anomalous advective heating. The right-hand panel, lower panel, is showing a schematic of the intermediate layer advective heat flux convergence that's contributing towards heating the intermediate layer. And this is also, the winter is also marked by the deepening of the bottom layer, as shown in the top left panel by the red curve, which is the averaged depth of the bottom layer, which is an isopycnal layer, and that demonstrates the propagation of the coastally trapped waves that deepens the isopycnal layers more during the El Nino anomaly, El Nino composites. And the upper right panel is showing you the strengthened poleward and meridional current anomaly, and that marks the strengthening of the CUC at the south face of the ESN. Overall, this indicates that this propagation of the downwelling waves, as well as the convergence of the fluxes, is disrupting the local memory that probably is providing more correlations in the ENSO-neutral years, and during El Nino years, that's disrupted by these waves. So that's all, and I'm happy to take questions, and also you can take a look at my uploaded online poster. Thank you.
I thought so.
Hi everyone, this is Jomi again. And time I'm going to present the work when I was a WHOI postdoc. It is about seasonal prediction of bottom temperature on the northeast US continental shelf, and which has been published last year. And here just list my co-authors for this work. As we know that the northeast US shelf is a highly productive marine ecosystem that supports some of the most commercially valuable fisheries. And different from the West Coast, recent studies have found that current global forecast systems have realized very limited prediction skill in this region. So our study is to investigate potential predictability using an alternative approach, which is statistical prediction models to predict bottom temperature in this region. The target region here, as shown in the upper left figure, is separated into 10 subregions, represented by different colors. And later we incorporated these subregions into the greater Northwest Atlantic domain, as shown in the right-hand side panel. And bottom temperature anomaly calculated for each of these shelf and slope boxes. They are considered as potential predictors in our prediction models. So for simplicity, here I'm only showing results of 3 subregions for the poster, which are Northern MAB, Deep Gulf of Maine, and the Northeast Channel subregions. And the data set we use for prediction is the glorious 12v1 ocean reanalysis data set. This is because observations near the bottom of the wall column on the continental shelf are still limited and discontinuous in time and space, which has been touched on by Susanne Wijffels in the last session. So based on this data set, we developed a couple of statistical prediction models. Except the local persistence and non-local bottom temperatures from nearby or upstream regions, we also consider several large-scale oceanic and atmospheric indices, for example, SSH-based Gulf Stream pass indexes and subsurface temperature-based Gulf Stream northward index, as well as the NAO index. And to avoid overfeeding, we perform a 12-fold cross-validation, and all prediction skills are assessed in ACC, RMSE, and Brewer skill score relative to the base model. Next slide, please. So here's the prediction skill map for these 3 regions in each row, with x-axis representing the forecast months during a year, and y-axis representing the lead time from 1 to 12 months. So the first column here shows the prediction skill of the baseline local persistence model, which is significant for lead time up to several months and varies notably by season and region. And the second column is for the persistence advection model, which relaxes the constraint on the choice of the predictor for itself to any box in the Northwest Atlantic. And the right 4 columns shows the prediction skill differences in ACC, RMSE, and the probabilistic skills in Brewer skill score for upper and lower terciles between the 2 models. So we could see that a significant improvement could be achieved by using the persistence advection model. And on average, the northern MAB box shows the largest improvement among the subregions, especially for October forecast and for lead times of 5 to 11 months, while for the deep Gulf of Maine, it shows the least improvement as its local persistence model already showed strong performance. Also, we found that most of those best non-local predictors that represented by numbers and alphabets in the figure are generally located along the upstream advective passes or within the neighboring subregions to a target region, which indicate possible advective signals. Next slide, please. So we also considered prediction skills from the large-scale indices, and among the 3 indices we tested, we found that only the T200-based Gulf Stream Index shows significant prediction skill, but the improvement were confined to some particular forecast months and were not significantly better than the skill achieved by the persistence advection model. Finally, to enhance the physical interpretation of these non-local predictors, we further calculated the prediction skill spatially for each subregion by treating each grid point as a predictor. For example, here in the right-hand side is a figure for the Northeast Channel region, and we could see that the region of best predictors gradually shift upstream along the slope with increasing lead time, and which confirms with our interpretation of the persistence advection model. So I'm going to stop here, and thank you for your attention.
Great, thank you,
All right, hello again. I just want to quickly share another couple nuggets of results for you. Some work that we've been doing at NOAA PSL to intercompare and evaluate the fidelity of high-resolution ocean reanalyses in the California current large marine ecosystem. The goal of this work is to get a sense for how well these higher resolution reanalyses perform at smaller and smaller scales. Our goal was basically to try and break them and see where they fall apart. So we do that by comparing to a variety of unassimilated and assimilated data sets. We tried to include as many independent ops as possible, but it is also nice to see how they compare to things that are in the structure of the simulation through the assimilation. So the 3 that we're comparing to are OARS-5, which is an ECMWF reanalysis product. It's a 1/4 degree. We're also comparing to what we call the California Current System Reanalysis, which is the UC Santa Cruz reanalysis that Chris presented earlier. It combines both the reanalysis component, which went from 1980 to 2010, but also we truncate the near real-time product to get a continuous time series up until near real time. And then we also compare to this 1/12 degree glorious product that as Wamin just presented as well. And we're looking in this domain here. So just, you can see on the left figure here, maybe I can show my cursor. Perfect. You can see the different observations that we are comparing to. Oops, spoiler. The different observations that we're comparing to. Throughout the whole study, we're looking at surface variables, temperature, sea surface height, mesoscale variability related to eddy kinetic energy. And then we look at temperature throughout the water column and then bottom temperature. So we're really focused, in this initial study at least, on temperature variables and sea level. Today, I'm just going to show you a couple of variables of interest based on some of the other talks that we've had today, and just show you generally how these different products are performing relative to them. First, we're going to compare to glider data. So these are 3 glider line measurements coming from Monte, Point Conception, and Dana Point. So glider lines 66, 80, and 90. So each row is a glider line, basically. And you'll see the annual mean climatology as measured by the gliders on the far, my right to your left. And then you're going to see the annual mean biases in each of the analyses, with hatching indicating significant biases. Each of the reanalyses have their different quirks. Overall, Glories has the lowest biases in this case. OARS5 has full column biases, warm biases that seem to be related to errors in upwelling rates, which is perhaps not surprising since it's the coarsest resolution reanalysis that we're comparing to. And then the UC Santa Cruz product has too deep of a thermocline, basically. So there's a thermocline bias that it's too deep, so you get this warm bias throughout the upper part of the column. So mean biases aside, we can look at variability. There we go. No, more spoilers. We can look at variability. So this is monthly mean correlations. On the right side, right column, again, you're seeing monthly mean standard deviation as measured by the glider products, and then the subsequent columns are showing you the point-wise correlation between water temperature at each point, monthly mean water temperature at each point from each reanalysis compared to the glider data. Stipples in this case tell you that the correlation is not significant. And overall here we see that Glories has the strongest correlations, the deepest, basically. But all of them are doing well close to the surface where they're more constrained by winds, and they're also constrained by satellite SST. But in the subsurface, Glories seems to be doing something more reasonable. Not to say that OARS5 and CCSRA aren't doing something good, that all of those correlations are significant that you're seeing for most of these, but Glories does explain more of the variability. And then very quickly, I just want to show you some bottom temperature results. These are comparisons for CCSRA and Glories to bottom trawl measurements, the same network that Sam showed in her presentation. Each dot here is a bottom trawl measurement, and these things are taken very sporadically, typically during the same time of the year, roughly, and they last on the bottom for about 10 minutes. So they're not meant, this correlation that you're seeing here is not meant to represent time variability, but it's mostly spatial variability because they're largely sporadically placed. I don't have a ton of time to get into the different colors here, but long story short, Glories, again, is representing bottom temperature more strongly than CCSRA, although CCSRA is doing something sensible as well. The main difference is the way that they treat bottom bathymetry. Glories has a terrain following bathymetry mask. CCSRA is a sigma coordinate, and there's the differences in where the bottom is in the model versus in reality. But one more thing here, if we want to look at time variability in the bottom temperature, we can stitch together a time series, monthly mean time series of bottom temperature from the glider lines, which are these top 2 rows, and the Newport line, which is the bottom row. And once again, we're looking at comparisons of the observations in black and Glories and CCSRA in red and blue, respectively. And you can see the correlations of how they're doing at each of these points. And once again, sort of the theme here is that they're both doing really well. Glories does tend to have the higher correlation at all the points. And overall, I just want to emphasize that these are basically grid point comparisons. And so even at the grid scale, these reanalyses are doing something sensible. So we really did try to break these things. We zoomed in as close as we could, and it's still doing something good. So I'll leave it there. It's about to give me the hook, but thank you.
Thanks, Dillon.
Oh, and sorry. And finally, we have Rafael Sevilla. Am I saying that? Savili. Thank you.
Thank you. So I've been starting my postdoc 3 months ago at the Jet Propulsion Laboratory. So today it will be more a general talk about my expertise, which is about the numerical modeling and remote sensing of the land-ocean aquatic continuum. So the land-ocean aquatic continuum is a mosaic of ecosystems, from terrestrial systems with river to the land-ocean interface with estuaries, coastal wetlands, tidal flats, and then it ends up into coastal waters and then the open ocean. And this continuum supports many ecological functions such as high productivity and carbon sequestration. A large amount of nutrients and carbon transits through the land-ocean aquatic continuum, to the open ocean. So one of the main questions that I try to answer through my different studies is to evaluate the productivity of coastal ecosystems, but also to evaluate the biogeochemical exports from rivers and coastal wetlands and estimate the global carbon cycle of the ocean, the sensitivity, of the global carbon cycle of the oceans to these exports. So to do that, I combine numerical modeling and remote sensing. Yes, that's good, thanks. So during my PhD, I studied tidal mudflats. So let's travel a bit in France, on the Atlantic coast. I study an intertidal mudflats on the Atlantic coast, and tidal mudflats are these huge areas covered by mud that's exposed during low tide. And as you can see on the picture on the left. And during low tide, one of the main primary producers on tidal mudflats is benthic microalgae, called also microphytobenthos. And these microphytobenthos are really important for the ecosystems because the waters are really turbid, so the phytoplankton production is not that high, and there is no macroalgae in these systems, so microphytobenthos support a major part of the ecosystems. And so we build a model system that simulates the physical conditions at the mud surface, so the mud temperature, the light, the tides, the waves, and the meteorological conditions. And we couple it with microphytobenthos models that simulates the biomass and the primary productions of the microphytobenthos. And we did several experiments with this model. For example, the figures on the upper part of the slide represent the simulated export of microphytobenthos biomass from the tidal flats to the water column. And we saw that the export is stronger during winter and fall because of the combinations of waves and a high microphytobenthos biomass in the sediments during these seasons. And we have been also able to simulate in 3D the microphytobenthos primary productions over the mudflats. And during my PhD, we also used remote sensing products, mainly multispectral images converted to vegetation index that inform us about the horizontal distributions of microphytobenthos biomass. And we also coupled remote sensing with modeling in order to map the microphytobenthos primary productions by using simulated feeds from the model to constrain the biomass detected by the remote sensing products. And finally, I want to finish by my current project, which aims at evaluate what rivers and coastal wetlands brings into the global ocean. And to do that, we will introduce nutrient river runoff and coastal wetlands exports into the Eco-Darwin model that Dimitris presented you before. So with these solutions, we'll be able at the global scale and the regional scale to estimate the contribution of rivers and coastal wetland to the RC CO2 fluxes of the global ocean. And finally, we'll be also able to evaluate the response of the phytoplankton activity and diversity to these exports. So if you want to go into more detail, and if you have some questions, I will be glad to answer you next to my poster right there. Thank you.
Now Dillon and I will lead discussion, right?
Right. This would be a great time if we had any questions that we had to push you off on because we ran out of time. This would be a great time to ask those questions. So, the floor is open for general discussion. Yeah.
I actually have a question to you. Yeah, so you saw some calculations. Is there any connection between your mobile signatures or that?
I'm sorry, can you rephrase?
So body heat fluctuations, could you have connections for the fundamental temperature?
Yeah, so if I understood correctly, was there a connection between the blob and bottom temperature variability? Yeah. We do see a bottom temperature signature related to the 2015-16 marine heat wave in the California current system. I don't know if that time series is the best way to see it, since it is very fine scale, point-wise time series that I showed there. But we are doing some other work to quantify bottom temperature extremes, bottom temperature heat waves, basically. And we very clearly see a strong bottom temperature signature. So stay tuned for that. Thanks for the question.
I have a question for Jake, actually. So it seems like one of the real challenges of IOS is that there's a very diverse profile of stakeholders that are looking to benefit from the ALPS. I was just kind of wondering whether you could describe a little bit who those stakeholders are, and whether they're sort of dominant interests or what the spectrum of stakeholders is.
Yeah, that's a good question, and I think you get 11 different answers if you had all of the RAs here. For us, and I should say the ICE Association, I think it's done now, or nearly done, has funded a survey through the Middlebury Institute for International Studies that gets at this. And it looked at user, who the users are, what they're getting, and why from the different RAs. For NERACOOS, which is, as we expected, the biggest group of users were different members of the private sector, primarily commercial fishermen, coming to us for real-time data for operational, navigational, and safety needs. It wasn't the only use. There were, we heard from government agency staff that they were coming to us for data for regulatory decisions, researchers, educators. So it was a pretty broad spectrum. But the biggest single group was for real-time operational data. Now, that may be, it may be an if you build it, they will come kind of scenario. Like we've had for 20 years a buoy network out there providing those data, and they've built up a fan base. Whereas in other regions, I think what Clarissa talked about, for example, they lean more on a glider program that serves other users and other needs. So I think that's what our user base I think looks like. But it does vary from place to place, and a lot of it is kind of in response to the systems that exist. The users find the systems as much as the systems find the users, I think.
Can I ask a brief follow-up? How much of the budget that you guys have do you find you end up spending on that primary user?
Well, I mean, it's hard to say how much on the user. It's more on the system. So our budget is about, I would say, close to half our budget goes directly into buoy operations. But those users are not only, it's not just the buoys, right? So there's, our buoy viewer provides NECOFS forecasts, and many users use that as much as they use the real-time data. It's not just what's happening now, it's, if you're a lobsterman, what's happening now is of interest, but what's going to be happening 4 hours when you get out there is of much interest. So the forecast system, the data management system, the data products, I mean, that all serves those users. But just buoy operations and some of the front end data management that goes with it is close to half our budget.
Thanks, yeah. Appreciate it.
I also have a question for Jake. You're popular. So I'm wondering if your organization as a kind of entity can facilitate in any way this idea of beginning to use UAVs in the coastal ocean, because it's a bit of a minefield as a Fed. I wear two hats. I have an affiliation with the university, and that's where I do all my drone work, because it's easier for me. I cannot wear my NASA hat when I talk about those. So I'm just wondering, too long don't read is, can you help facilitate development of those types of systems?
That's a really timely question because, again, with just the history of our system being so built around the buoys and that being such a priority, in some ways it's gotten in the way of diversifying and expanding. And there was a campaign over a couple of years that resulted in new congressional investments in radar as well as gliders. That let us start to do a little more with those systems, but it's been hard to really grow and keep it going with funding where it's at. I wonder if funding crunches continue, if it actually may force us to go in that direction. Particularly when it comes to, I have trouble seeing anything replacing the value of the coastal buoys for those operational needs. But as far as kind of the more offshore areas, and this is offshore on the shelf, right? So this is the deeper basins of the Gulf of Maine out toward the Northeast Channel. For the science that those couple of offshore buoys are producing, I wonder if we might do it cost effectively and maybe in some ways better by using UAVs. I don't know. But we are. This is something that Rue got going a couple of years ago. Shortly before he retired, he held a meeting with all our operators. Just, one of the big questions was, we've got new money for gliders. What are we going to do with them? What's our glider plan? What's our strategy? And then between his sickness and the pandemic, that question never got answered. So we're picking that back up now and developing a glider plan. But I'm starting to think it maybe should be more of a broader UAV plan.
Because we have spoken for years and years in the ocean cover community about the utility and huge potential of UAVs for the coastal ocean, for all the reasons that I spoke about there. The time is, economies of scale now. I bought a fantastic drone that would have cost me like $100 grand not that long ago for under $5,000 with a 5 kilogram payload, and it's just ridiculous what we can put on them.
And doing it more cost effectively, of course, helps. But I'll make one more comment that I made in a couple of my breakout sessions, that I think getting those to applications is pretty key as well, because when we go to Congress to beg for money, if we have a lobsterman sitting there right with us saying, yep, every morning I get up and I have coffee with NERACOOS when I check my buoy and decide if it's safe to go out, we need someone to be able to say something similar about those systems. And maybe it's not data that are directly coming from the UAV to their laptops, but at least if they understand that this is fed a forecast model that helps them know where to fish or when to fish. So I think the data to model to use the application linkage needs to get stronger. But having it be cheaper will certainly help.
Yeah, and my instantaneous response to that is chlorophyll for fronts for fishing and halves. A lot of the halves, a lot of the toxin-forming halves do have a very distinct optical signature. Corellia brevis is the one in Point Alexandria, a little more challenging, but those are low-hanging fruits.
I agree.
Susanne, sorry. I was really curious about, you mentioned that you incorporated known unknowns and unknown unknowns into your model. I was so curious about how you do that, but other people have experience doing it, so I was really curious.
Great question. So I'm gonna preface my response to say I'm not the data scientist. My colleague Erdem and Ian are the data scientists, but there are. In fact, we were in the same breakout session, and I think we were speaking about this the other day, and I pasted it into one of the breakout sessions. There's a really nice overview article on how the known and unknown unknowns, how they get represented in a Bayesian framework. So there are techniques out there. It's the famous Donald Rumsfeld quote from many years ago, and everyone laughed at him, but it's actually a thing. But maybe offline I can send you the link, and we can discuss more with our data sciences colleagues. Great question.
Someone puts a great online Go ahead, Al. You can.
So here we go from Al Herman. Oh, is he going to speak?
Yeah, let's see.
Yeah, I'll just read the question I just typed in. Yeah, this is directed to both Dimitris and Chris. I assume all you have really is like satellite chlorophyll data to assimilate, maybe some in situ as well. How, when you're assimilating that, how do you apportion that into different size classes of phytoplankton? For the Darwin model in particular, it's legion for having multiple size classes of everything.
It's true that all we have for assimilating into the biogeochemical model is surface chlorophyll at the moment. And what we do is we partition based on the uncertainty estimates in that error covariance matrix. So how we have a prior estimate of each functional type, and we have some uncertainty associated with that, and then the assimilation procedure partitions adjustments to both of those within the uncertainty estimates that we give it, so that the total matches the total chlorophyll. But there are lots of sources of uncertainty, one of them, for example, being the carbon to chlorophyll ratio for the functional type as well.
Like maybe my question is even simpler. Say you've got X concentration of small phytoplankton and 2x concentration of big phytoplankton at the moment that you're assimilating. So you divide it based on that instantaneous amount, relative amounts of the two size classes?
So if you have X plus 2X, you first convert both of those to chlorophyll, and then you look at the difference between the observed chlorophyll value and that sum that you have. And then you calculate adjustments to both phytoplankton types such that you get closer to the observation than you were before, but the amount that you put into each is dependent on their relative uncertainties. So you might adjust one a little bit more than the other based on how certain you are of your background.
Okay, but still influenced by the. Oh, sorry, I'm getting an echo.
I'd also like to say we're working on a more sophisticated way, which is to assimilate wavelength-dependent information that will be more phytoplankton type specific. And I'm sorry, I just said the wrong thing.
Oh my God!
So this is Dimitri. Just have the answer is not yet. It's a Blair Witch project for us. So far, the only thing we have assimilated in a very low-dimensional way is biogeochemical tracers and DIC. And we have adjusted some biological parameters, but they were not. First of all, our ecology is a very simplified version of Darwin. Stephanie picked 5 phytoplankton and 2 zooplankton. So we haven't yet started to do that. However, we now do have a funded project that's underway that's going to start to explore those questions, more complicated ecology. The Darwin now includes radiative transfer code, so we're hoping to constrain directly using reflectance and ocean color and pace observations when they become available. But that's still Blair Witch for us project, hasn't been done. I love that term.
Okay, thank you both.
I wanted to ask Dimitri a question too. It's really just a yes or no question, but the beauty of having a 40-bar adjoint representation is you have dynamical consistency, and you can thereby evaluate balances and determine what terms are contributing to the changes in variables. If you save all the terms properly integrated over some time scale, you can't just keep every 15-second version of the term. So over what sampling rate are the integrated budget terms saved? Monthly or?
I mean, we save them monthly. However, we also give the code to people, and they could run.
I see. You just rerun the model one time.
They could rerun any section of the model and save anything they want. That's kind of our approach. But what's saved is mindful. So it wasn't a yes or no question. Yeah.
Susanne Craig a bit of a mystery today when she was presenting her work for drawing the World Ocean Database. I've done that in the past too, and I've always been a little surprised at how little data I could pull out from that, because having been familiar with a lot of cruises been down in the coastal regions over the years, and I'm kind of wondering how much of that is actually that we haven't been out there observing, and what fraction of that is that they just haven't made it to the World Ocean Database. And I'm not sure if Susan can answer this best or somebody else, or maybe Chris can, because he has done that.
Yeah, so to do the type of work that we go to actually lots of different places, and I think there are a lot of pieces of information that are not in the World Ocean Database. Not, the glider data is in one place, and some of it is in the World Ocean Database. Other pieces of information are more distributed. I think they get in eventually, but maybe 2018, 2019 is too much time.
That's definitely a latency would want to remove, if possible, if we're going to use these for predictive and recent state estimation purposes, which is when arguably they're most useful for a lot of forecasting.
Yeah, we don't bother with World Ocean Database for near real-time stuff. We really want to go to data collectors as quickly as possible to get beyond this.
So you have to sort of develop your own network of collaborators on the data stack, have the personal contacts with those collaborators.
They're publicly available, but it actually takes a lot of time to collect all of the data that you want, and then as soon as you're done collecting it, suddenly names have changed or site interfaces have changed, and you have to start again on that particular data set. So it is a lot of effort.
A follow-up question, maybe for Susan, or anybody else that wants to chime in. Only seen a few little pieces on tagging data where we have a large charismatic megafauna carrying around CTDs and also maybe oxygen sensors and some things like that now. Is that an untapped resource? Is that an untappable resource? Can Argo or an organization like that serve as the standard or a steward for those kinds of data, or am I kind of naive about how useful they might be?
So I think there is a lot of tag data already well curated. There's some great international projects doing that. And they do turn up in WOD, they turn up in the European version of that called CORA. So there's a lot of animal tag data actually, because there is an international process. There's people working on it, curating it. I don't know about the oxygen part of that very much, because that's not a variable I've looked at a lot. But certainly the CTD data are there, and they're awesome because they are operating in areas where platforms like Carlo and other platforms are not operating. So they're really good. Yeah.
Stakeholders care about. Some of them care about those areas the most because it's associated with fishing or watching tourism.
Yeah, so I only really know about a part of that. I don't know again how globally complete the activity is, and it would be great if it was globally complete and the data were all being curated in the same way. I'm not a true expert in that platform.
I agree with all that, but there's a lot of tagging data going on, and I think a lot of progress has been made in taking what were once very siloed and disconnected projects and bringing them together. But to your question about those critters carrying other sensors, I think it's untapped but not untappable. There has been some work done. There was at least one paper that came out a couple years ago on kind environmental sensors, basking sharks, which is kind of cool. But I actually think the better source of that type of, almost crowdsourced data is ships of opportunity, particularly commercial fishing vessels. There's a good project run in the Northeast out of this Jim Manning at the Science Center and Aaron Pelletier at the Gulf of Maine Lobster Foundation that started with lobster traps, but is now getting environmental sensors on trawlers and long lines. And that's a really nice source of, originally mostly I think temperature data, but they're starting to add and develop, and they're contributing to developing smaller, lower cost sensors. And so that's a way of getting kind of opportunistic measurements all over the place that I suspect is going to be a more useful and cost-effective approach than sharks and dolphins. We do have tags on sharks though.
There's a lot of money for them too.
They're not always cooperative.
Mine's on red.
Okay.
So getting back to all the talks this afternoon, what were the things you wanted to ask that we didn't have time for? Ask the speakers now.
No one else did, right? If I'm not going to do anywhere from there, I feel okay about this. So one point about the data that I haven't found that we've discussed yet is, for predictive purposes, there's been a lot of work on simulating chlorophyll, and I think we have the observations and it's useful, but chlorophyll, of course, is very exposed to the atmosphere, which is noisy, chaotic, and I guess I'm really excited about the subsurface information, both because it has a longer memory that being isolated from that to propagate signals, and because Chris probably appreciate it, the covariance structure is probably a little bit simpler in terms of assimilating. And Chris, have you guys had a crack at the DCR or anything like that? How have you found your success with the subsurface relative to the surface?
We've recently been assimilating collider fluorescence chlorophyll, and we're, I would say, equally successful with the subsurface information relative to the surface. We did run into some technical problems where we reached the bottom of the fluorescence estimate. We didn't realize it, and so we had unrealistic estimates for a while at 1,000 meters. But anyway, with some quality control and some understanding of the limitations of the instrument, we were able to assimilate. I think where the exciting stuff comes is in assimilating other variables: oxygen, PCO2, pH, nitrate. I think are going to have subsurface nitrate is probably going to have the biggest impact on our work.
Oh, sorry. Can I just ask a follow-up question on on the chlorophyll from fluorescence? Forgive me if you know this already, but there's a photochemical quenching near the surface that suppresses the yield that's accounted for. Yeah, that's a known phenomenon.
Yes, that's a known phenomenon, and that's accounted for, but I'm suddenly 50% sure of what I'm saying.
One way to get around it, and what I've done on gliders as well, is just use nighttime data, middle of the night when the photochemical quenching is relaxed.
So I guess what we do is we tend to trust the instrument people to give us the information. We don't create the chlorophyll data ourselves, so we're trusting that it's accounted for the way that you're suggesting, but I really am not sure.
I'm hoping that they do. It's a well-known phenomenon from the 5 nitrogens. Just a rabbit hole.
We found some mismatches between the fluorescence-based estimates and the satellite estimates. So we do try to
do some readjustment of the fluorescence profile based on that.
Thank you.
I wanted to make one other point about data assimilation in the 4D bar sense, where you're doing it in a dynamically consistent way. You know, all the work that these guys and others have put into producing these long-term reanalyses is really important, but you can also, as long as you have either really smart friends like Chris Edwards or really great students like Ha-Jun Song, you can use data assimilation to try to diagnose things on much shorter timescales at much finer resolution to try to understand how physical processes control ecological processes. And in the context of the California Current Ecosystem, LTER, Long-Term Ecological Research Site, they go out once every other year and spend about 4 weeks at sea measuring different rates and things between organisms, grazing and growth and all kinds of stuff that you would not normally get from things like coffee. So now they have and they do it on around, say, an eddy or a filament or a front, right? Now they have this highly resolved ecological behavior over a short time period. And we've done things with Ha-Jun, particularly leading that, my former student, where we use the 4D bar framework for about 6 weeks, not stopping at sort of the 7-day Chris Edwards limit or doing it for 10 years like Dimitri does. So just for 6 weeks. And so you can then adjust the forcing and the initial conditions and the boundary conditions to come up with the best guess, fully evolving vertical velocity field, lateral advection field, subduction processes, and people then like Mike Stugel, the paper was cited either earlier today or yesterday, was able to show that the particulate sinking that they were observing was actually being facilitated by subduction processes in this eddy. So there's a really scientifically interesting, tailored way to use these fancy techniques of data assimilation to get at things that if you didn't have the capabilities of the modeling, the assimilative strategies, and the highly resolved biological novel measurements, you would never have been able to get that type of a result. So I just wanted to point that out. Didn't you go?
Yeah, I actually have also maybe a question, an idea, or something to reflect upon tonight. So we have had this great presentation on data simulation and observational capabilities, maybe prospect of integrated data set, or maybe like the idea that Susan put forward, but maybe like a backbone of coastal or maybe global observation, but I stick to the coastal for the time being. So how can then we integrate, so suppose we get there, how we would envision an integration with the modeling component? We have discussed a little bit during our breakout today, but how would that inform the modeling? What, and actually I'm looking at Charlie. So how would you envision like maybe integrating this richer and more sustained or more organized data sets with coastal modeling or maybe global modeling more resolved along the coast?
Have a crack at it.
Yes.
I think the last couple of comments have illustrated a couple of ways. Art just said, of course, there's one great way that these feedback is mapping out of those, those key challenges that we talked about earlier today, which is understanding, mechanistic understanding. That key challenge is connected with both modeling and observations because it's really the integration that'll help with the mechanism. So I think that's one important piece of the puzzle for the mechanistic understanding of these interactions. I think Chris and his work is really blazing the trail at that WCOP system is the first snapshot of what could be eventually a national system of these real-time ocean state estimation and short-term prediction for operational use. And the Ecocast example is a great one for that. I think that the payoffs of that will continue as one moves up further in time with examples like JScope. What I think we got to do is really figure out how to extend the sort of examples that Chris shows and extend them to all systems and extend them out to longer time scales. And I think it's going to be all hands on deck approach to that, where, I think the short-term infrastructure, NOS is doing a great job through projects like Chris to build that up. I think we can bring the climate change elements, things like what Liz was talking about today, which are less dependent on the assimilation, perhaps, but very much dependent on the mechanistic understanding and on the ability to validate the models that you're going to mechanistically project into these novel environments, right? So really, that robust data set on climate timescale for multiple decades is really what we need to build up for all those applications. Hopefully that was coherent.
Unless anyone has any closing statement, I thought that was a perfect closing statement by Charlotte.
Yes, from the book.
I think it's been a long day. We can end 8 minutes early if it's permission.
Let's thank all of our speakers from earlier.
Yeah. See you all tomorrow.
Yes.
Or tonight in various places around town. But I mean, when I was originally asked, I said I would do it, and then I got it.